Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. Climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research. Scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. Of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers. 2. Accrediting scientific journals. 3. Studying and evaluating biodiversity. Establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO and has 3 international centers, namely Asia-Pacific Centers for Eco-Hydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future. Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967 but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research. Scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world.
Ladies and gentlemen, we will start the International Symposium with warm welcome remark from the Chairman of Indonesian Institute of Sciences, LIPI, and also will be officially open our symposium. Dr. Laksana Trihandoko, the time is yours. Thank you, Pak Handoko, for the remarks. Ladies and gentlemen, the next agenda, welcome speech from the Deputy for Engineering Sciences, LIPI, Dr. Engineer Agus Haryono, the time is yours.
Welcome, ladies and gentlemen. Thank you to each and every one of you for being here virtually today at this important, the 6th International Symposium on Chemistry 2020. My name is Agus Haryono, Deputy Chairman for Engineering Sciences of Indonesian Institute of Sciences, LIPI. It is such a great honor for me to speak on behalf of LIPI. Let me begin by giving you a warm welcome to all scientists and scholars from all over the world to take part and deliver their ideas to this virtual conference. Before we get started, I would like to express my appreciation to the Honorable Chief Guests, respected directors, speakers, and all our other guests. We could not have done this conference without your participation. I am also grateful to all the 6 ISAC steering committee members, the organ, and all the volunteers who have arranged this program. I will kind-heartedly welcome all the participants from academia, students, industrial practitioners, and researchers. We are also fortunate enough to get supported by our partners, whom also organized this scientific conference. This conference is a part of Indonesian Science Expo 2020, aimed to bring together researchers, academians, and industry to share information, to share knowledge, and also their research results. I do hope that this conference will serve a platform to provide solutions to the problems on natural products and traditional medicines, pharmaceutical chemistry, pharmaceutical technology and drug delivery systems, analytical chemistry and chemical engineering, and also food technology and functional food. I wish you all a fruitful and productive discussion during this conference. Last but not least, I would like to sincere thanks to all of you for being able to join this virtual conference. And please stay healthy and happy during the long pandemic. Again, thank you very much and enjoy the conference.
Ladies and gentlemen, also we will hear the speech from the Chairman of International Symposium on Applied Chemistry 2020, Mr. Abdi Wirasiptama, PhD. The time is yours.
Very good morning, ladies and gentlemen. I'm Abdi Wirasiptama, Chair of ISAC 2020. Welcome to the 6th International Symposium on Applied Chemistry 2020. This conference was organized by Research Center for Chemistry, Research Center for Biotechnology, and Research Unit for Natural Product Technology, Indonesian Institute of Sciences. The aim of this conference is to gather academic, scientists, and professionals from several disciplines of applied chemistry to share the knowledge, innovation, and experience in developing innovative products and technologies that address challenges in the field. Papers have been received in order to be presented in ISAC and will be published in IOP Conference of Material Science and Engineering Index in University. Professor Dr. Nina Artanti from Indonesian Institute of Sciences and Associate Professor Nurul Huda from University Malaysia Sabah, and also the organizing committee of ISAC 2020. We are also grateful to all support and suggestions that come to us. And finally, thank you very much and have a great conference.
Our honorable guests, plenary speakers, moderators, and all of participants. Before we start the next session, it's better for us to take screen capture for our documentation. So please all of the guests and also all of participants, turn on the video. The committee will take the capture screens. Okay, 1, 2, 3. Well, once more. 1, 2, and 3. Thank you. Ladies and gentlemen, there are several announcements to all of participants. First, please have a stable internet connection during our symposium and make sure that your computer has a working camera and also audio device. The host will automatically mute the audio all of the participants. And the chat room is only available for delivering questions to the ongoing discussion in the session, requesting for changing or moving to the other breakout room in the parallel session, and also announcement delivered by the organizing committees. Dear keynote speaker, moderator, and also all of participants, please don't forget to fulfill the list of attendees that the link shared by the organizing committee in the chat room. And please properly rename your Zoom ID according to your participation category to set you up to the correct breakout room in the parallel session. For the participants that want to asking question, please state your name of institution and also the panelist name that you want to deliver the question. Well, ladies and gentlemen, from now on, we will start the plenary session 1. In the plenary session 1, we have 3 speakers. Professor Dr. Gunawan Indrayanto from Universitas Surabaya, Indonesia, Dr. Rahmat Ambara Rahmat Pradipta from Tokyo Institute of Technology, and Professor Dr. Muhammad Iqbal Chaudhary from Karachi University, Pakistan. This plenary session will be moderated by Professor Dr. Andrea Agusta. Professor Dr. Andrea Agusti. Well, ladies and gentlemen, I will give time to our moderators. Professor Dr. Andrea Agusta, please take your time.
Okay, thank you very much for fourth time to lead the plenary session in this morning. So I already sorry. Good morning and assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, everybody. So from now until the morning, we will have a plenary session, and we have three distinguished keynote speaker is already introduced by organizing committee, but I would like to introduce more detail about the speaker one by one. We have three speakers, and each plenary lecture will be held about 30 minutes. And then in the end of this session, we will have also 30 minutes for discussion. So during a keynote speaker presented their works, you can ask everything via chatting rooms, or yeah, but I cannot find this. If you want to deliver a question directly, maybe you must raise your hand by a signal in this session. Okay, I would like to introduce the first keynote speaker, Professor Dr. Renat Gunawan Indrayanto. Current position is professor at Faculty of Pharmacy, Surabaya University, Surabaya, Indonesia. Latest education and training of Professor Gunawan is Dr. Renat magna cum laude in 1983 at the University of Tübingen in Germany. He ever worked as a visiting researcher at Leiden University in 1989. Also in Düsseldorf University in 1994 till 2002. Presently, his main works are analytical method development, including chemical and bioassay, and its validation, metabolite profiling, and also natural product chemistry. Professor Indrayanto currently has 120 international publications, consist of original article, short communication, and review article. His publication H10 index were listed in Scopus, also ResearchGate, Google Scholar, Sinta, and some other database. So I would like to ask to Professor Indrayanto, already ready to deliver your presentation in this morning? Sorry. Okay.
Okay, good morning. Thank you for your nice introduction. First of all, I would like to thank to the organizing committee for the nice invitation. It is a big honor for me to share my presentation in this occasion. Distinguished participants, ladies and gentlemen, I will share my slide first. Okay. hope you can read this slide. Can you read this slide, Professor Abusar?
Yeah, okay.
Okay, thank
Let's
you.
go.
Yeah, thank you. The title of this today's presentation is The Importance of Validation Method in Herbal Drug Research. Herbal drugs, also sometimes calling botanical or phytomedicine or herbal medicine. Herbal-derived drugs usually consist of very complex chemical, sometimes mixture of natural products, and often containing multiple herbal or plant. It is very common a single herbal product could contain many, more than one plant or herb or extract. We know that herbal drug is very important because extract and phytopharmaceutical, but non-standardized jamu is also available. Around 75% of Indonesians also consume various types of jamu. We know that there are countless scientific publications on herbal drugs or medicinal plants, but unfortunately, most of the marketed herbal drugs in Indonesia are not yet fully scientifically established regarding their safety, efficacy, and consistency best to best. This is the reason why the application of these herbal drugs by medical doctors in Indonesia is very limited. I think it is also maybe in other countries. Many published work on herbal drugs or medicinal plants did not report their method of validation completely. This means the validity of the conclusion cannot be confirmed. As a consequence, this publication have only limited value. For developing a modern drug from herbal or plant, chemical and biological assay are always required. So the availability of validated method is very crucial. Without applying validated method, all conclusions may be wrong or biased. We know that most of the Indonesian traditional drugs consist of mixture of plants and or their extract. Indonesia is also very rich on natural products, plant, animal, marine biota, and all of these natural resources can be used for the new drug development. Therefore, the resources of herbal drugs or plant or other Indonesian natural resources are very important in order to develop drugs for the drug self-sufficiency in Indonesia. This is the diagram of ethnopharmacological study plan. From traditional drug application, also jamu or others, usually we can do literature review and then evaluation the literature and deciding the plant material to be tested, designing the methodology, and after that we can collect the plant. After we can get the plant, usually we do the toxicity, bioassay, safety, and also phytochemical study for preparing standardized extract and or pure compound. And then we can do some in vitro study to confirm the bioactivity. And if it is okay, and then continue to clinical study. And then after that, we usually do toxicity study of preclinical study and clinical trial. This is the same also. All drugs can be only marketed if clinical trial and all trials are okay. From that, I can make summarization of the important aspect how to develop modern drug from traditional drug. At first, we should collecting and authentication of the blood material. Can be used microscopic DNA profiling or metabolic profiling. And then predicting, determining the possible active compound or toxic compound and their chemical structure using chemical and biological method. And then determining the compound that can be used as marker for quality control purposes. Determining the detection limit of the contaminant of the toxic compound. Sometimes we need to isolate the active compound to determine the structure, especially this is for unknown compound. And then we can do development of standardization of extract and stable herbal drug for marketing purposes, and then clinical study of this drug. If all is okay, I previously stated. To have reliable data of characterization of plants and phytochemical or use chemical and biological method must be validated. This is the first, how to determine the active toxic compound and its chemical structure. This is two methods. The first one is using what we call bioassay guided isolation of active compound. In this method, the raw material, the plant, will be extracted with some solvent, maybe ethanol, and we can use some method, say, or solidification, and then after that, all of the extract will be tested their bioactivity. Only the active extract can be continued to fractionation with some method, and then only the active fraction can be further worked to isolate the pure compound. After we get the pure compound needed to be tested, and then the chemical structure of the active compound will be determined. This is the first method, bioassay guided isolation. The second one is using combination of liquid chromatography, mass spectrum, and multivariate analysis. By this method, the active metabolite or the active compound can be directly determined without any isolation. Something like this, we have profile MS, LC-MS, we have profile bioassay, and then using multivariate, we can determine which sample are active, and then using export, we can also determine which peak are active. Before we use this PGA or multivariate, we should first validate the method. And for the active metabolite, we can predict the starter, we can predict the structure using database or using authentic standard. This is the method of validation that published by Wan et al. This included all quality control sample or validation sample must be shown a tight clustering in the observed score plot, and coefficient variance of the described variable should be less than 30%. Variable show more than 30% should be removed. This is the example. This is show relative tight clustering, so it means the method is valid and can be applied for other purposes. This is the step taken for predicting chemical structure using database. At first, we should, by LC-MS, we can determine the exact molecular weight based on active. Active should be confirmed by extract ion chromatogram, and then we can determine the most probable molecular formula using isotope pattern, and then we can determine and confirming fragment ion based on observed mass spectrum data and their extract ion chromatogram. After we get all this data, we can insert to some database, and we will get a predicting structure and fragment, and we should evaluate it fragment, and that's it. We can have here LC mass spectrum, and from molecular ion, exact molecular ion, and we can get isotopic pattern. From that, we can determine the probable molecular formula. If we have all data used by molecular ion, this formula and the fragment of MS2, we can insert into some database or one or two database, and the database will predict one or two more compound. That's why we should confirm with other database. Or database, and we can confirm the structure using other database or publication, and evaluate the database using official. We know that all organic compound spectrum is 1, so for fulfill 4, we need 4. But if we have LCMS-MS, it needs only one precursor, Ip1, and 2 daughter ion. So the total SP also 4. But for high resolution mass spectrum, we just need one precursor and 1 daughter ion. But the mass accuracy should be less than 5 or 10 ppm. This is the same guardian from FDA 2015. This is the guardians. This is the difference that they describe the extra ion chromatogram retention time should be not differ 2.5% or not more than 0.5 minutes. And the criteria below is the same. If for only mass spectrum, because it is high resolution, we just need 2. 2 is 2 times 2 is 4. But if mass spectrum IP is 5. But if combined with mass spectrum and mass spectrometrum, it needs 1 precursor and 1 daughter ion. The IP is 4.5. This is the guidance. If we have IP at least 4, it means that the identity of the peak can be confirmed. So I will continue to quantitative determination of group or compound that can be used as marker for quality control. But I just only discuss the most important aspect of the validation method due to the limited time. Ideally, chemical marker for the purpose of quality assurance should represent the chemical identity profile and reflect relevant pharmacological and toxicological profile of the herbal medicine. Unfortunately, there are currently no detailed accepted current official guideline which can be used to select this chemical marker of choice. The challenges in assuring quality control of herbal products are biochemical, usually primary and secondary metabolite composition in herbal is very complex. So the chromatography is the method of choice. Many external factors can affect the biochemical content. So this is the factor that can affect the chemical content of a plant. This is plant species or plant variety, site of cultivation, age of plants, and kind of organs, season and time harvesting, post-harvesting, method of drying, method of cut, extraction method, and formulation, fabrication, active. This all can affect the biochemical content of herbs or a medicinal plant. So it means identical herbs could have different bioactivity due to herbal marker ranking system. Each of marker will be ranked as X 0, 1, 2, 3, 4, and 5. This is the ranking from Ben Susan. Marker X, it means there are no bioactivity study currently available for this compound. 0, compound activity is not related to indication of the disease interest. Disease, however, there is only one good quality studies to support this action. And compound is presently in low concentration between 5 to 50 microgram per gram, and the traditional or current use of this herb consistent with the bioactivity or pharmacological effect of the compound. 3 is this same, the difference only that is the same, that the concentration is higher, more than 50 microgram per gram. 3 is same with 2, the difference only the marker available in high concentration. And number 4 is same with 3, the difference that there are 2 good quality study. 2 good quality study, that is quality study about the first effect and clinical studies. And ranking 5 mean same with 4, but the compound have already bioavailable. It means they have already pharmacokinetic study of the compound. Or number 5, the compound may be toxic and they need to be screened or to comply the safety limit, especially for the compound that genotoxic or carcinogenic, that the the maximum permitted level is 1.5 microgram per day. In 2017, Professor Langer from Austria described a paper regarding this matter. I will summarize. Nearly all monographs on herbal drugs or herbal drug preparation currently in European pharmacopoeia contain an obligatory assay of a certain compound or a group compound and define the minimum content. This is also described same thing in USP 43, 2020 or Indonesian Herbal Pharmacopoeia 2017. Generally, they do not describe the maximum content. This is different if we see the monograph of chemical component. Chemical compound should always describe the specification ring of the product. In most cases, that obligatory is inappropriate for herbal drug evaluation, as the assay constituent must be considered as purely analytical marker without correlation to their efficacy and safety. It is nice to initiate the scientific discussion in order to adapt current quality standard for herbal drug and herbal drug preparation to reflect current scientific knowledge and requirement that are appropriate for quality control purposes. It means a certain chemical marker or compound that correlated to its pharmacological action must be determined and specified. This is important for quality tool. So indirectly, Professor Langer confirmed the work of Susan in 2015. In 2017, this is the guidance of WHO according to marker. Describe something like this. Marker for quantification should be represent the main therapeutic or pharmacological profile of the herbal material and or finished product. Marker for identification should be specific for one plant or for certain plant, species, or genera. If it's not, other markers should be selected for specific identification. They should be detectable and quantifiable in the product. Different substances must be selected for the same herbal material depending on the different form of the herbal preparation. Maybe this is aqueous or alcoholic or tablet, etc. or different therapeutic indication. A group of substances may be selected if one is not sufficient to evaluate the herbal marker of finished herbal product. This is the guardian of the WHO. In 2017 also, some researchers described what they call quality marker. Quality marker can be defined in the following condition. Quality markers should be existing in herbs, extract, herbal product, formulation, etc. Quality marker could be analyzed using qualitative and quantitative approach. It can be issued the authenticity or for differentiating the quality of the same herbs. The drug effect, such effectiveness and safety should be demonstrated to be associated with their quality marker. The quality marker is chemical substance that is traceable in whole process of production and preparation of the herbal drugs. So if we read this all key marker and guidance of the WHO, key marker is about the same meaning to herbs marker ranking system 3, 4, and 5 or WHO or WHO guideline. So only key marker or herbs marker ranking system 3 to 5 can be applied as a key tool for evaluating the quality of herbal drugs. Unfortunately, quality marker is not yet described by current pharmacopoeia, and only a few quality markers have been determined and published. If quality marker is not yet specified, it is recommended to apply metabolic profiling combined with multivariate analysis as a key tool. For having reliable data, the method of analysis of the key marker should be first validated. Its selectivity, linearity, accuracy, precision, detection limit, quantization limit, etc. And this is the summarization of the recent development of chemical marker and metabolite profiling. If you're interested, you can send email to me. I will send you the full paper of this two review article. I will choose the most important. This is the first as the selectivity. As I said previously, due to the complex nature of herbs, chromatography is the method of choice for this method of analysis. To prove the selectivity, the target peak, this target peak should be proved regarding its identity and purity, because identical retention time does not mean identical compound. One peak does not mean a single compound, so we should use direct ray detector or mass spectrum to confirm this peak. But the problem is different compound maybe have identical UV vis spectra. So mass spectrum is better detector, but mass spectrum is the best, as I described previously. So the second important is how to express the linearity. Usually, to express the linearity, we using correlation coefficient. But correlation coefficient cannot be used anymore as whole parameter. As you see in this slide, correlation coefficient R square 1, almost 1, the error could be 1%, R square 0.987, the error could be 4%. So the correlation coefficient can be used as a sole parameter for evaluating the linearity. Actually, this has been published in 8080 by Association of Chemical Analysts of USA. So for that purpose, we should use other parameter. This is some parameter that can be used to express the linearity. The easiest one is use correlation factor, and the ANOVA test. ANOVA test can be performed by Excel. So it means that correlation coefficient, if the calibration is okay, correlation coefficient is okay. But the correlation coefficient is okay, it does not mean the calibration is okay. Sometimes if we work with other common internal standard. But before we can use this equation, we should do linearity of all target compound, and common reference standard must be evaluated, and identification of the target peak is performed using relative detection time. This is the criteria if we want to use this equation. So by this method, we can determine any marker, any compound without using other internal standard that's more cheaper. Because in many pharmacopoeia, they always describe the minimum content of total phenol and total flavonoid. I assume the publication of Matic at 2017. Spectrophotometer method for determination of total phenol and total flavonoid were generally used because that is economical and simple. But the obtained results were different for both methods, which use different polyphenol or flavonoid standard. And the difference is statistically significant. So the total polyphenol and flavonoid in the sample must be determined according to the standard that is found the most abundant in that sample. That is why selecting the correct standard is crucial. We cannot use any standard for determining the polyphenol or flavonoid. We should know what is the most abundant compound in that sample. And of course, the method should be validated. So I will continue.
Sorry, Prof. You have 2 more minutes.
2 more minutes. Okay, and then 2 more minutes. This is the method of how to do the limit of detection using USB 43. This is how to make the sample detection limit, and this is how to validate the biological assay. For the complete discussion, you can read our chapter. The important thing is all cells should be first authenticated and the stability of sample should be evaluated. This is the cell that can be used, not all cells, but certain cells according to certain guidelines. So this is usually something like this, and we should determine the Z factor. The Z factor. This is the equation of Z factor. So the Z factor should be not less than 0.5. So the system cannot be used. So other methods should be used. Many publications that reported bioactivity, they do not reported this validity. So it's mean the validity of the data can be not confirmed. So that this may be biased or wrong. This is also should be determined the selective index. Selective index should be as high as possible. If selective index is below 1, it means that it's not acceptable, although the compound or the extract is very active. For continuing the work, we should do accurate potency of the test sample must be determined. So I recommend use the European Pharmacopoeia 2020 section 5.3. That is better. And this is the conclusion. Indonesia is very rich of traditional herbs and alternative resources that can be developed into phytopharmaceutical or modern drug. It is recommended to determine the quality marker and its specific strength for each of herb monograph that described in Pharmacopoeia Herbal Indonesia. Performing clinical trial for all marketed phytopharmaceutical is needed to have consistent quality of every batch of the product. For having reliable conclusion, all used chemical and biological assay should be validated. So, due to the limited funding and facility, I recommend we do some collaboration between all scientists in Indonesia to save the funding and to use the facility that we do not have. At last, I would like to acknowledge to Dr. Abdi Wira Saptama for the nice invitation, and thank you for your nice attention. Sorry, I will present at last of my presentation in very fast because the time is limited. Thank you very much.
Okay, thank you very much, Professor Gunawan. We have already saw the prominent result of your work concerning herbal medicine. You already present the one most important aspect in herbal medicine development, especially for quality control of quality of traditional herbal medicine. Okay, audience, we will have discussion in the end of this plenary session. Please keep your question until the discussion session. Okay, we go to the keynote speaker too. Dr. Ambara Rahmat Pradipta already joined in this room? Okay. Dr. Ambara, current position is assistant professor in the Tokyo Institute of Technology from 2006 and 2008. And latest education and training, graduate from Osaka University, Graduate School of Science, Department of Chemistry for PhD degree, and also conduct the postdoctoral research at Osaka University in 2011 and 2012, and also postdoctoral research in RIKEN from 2012 and 2017. Presently, his main works are bioorganic chemistry, biorelated chemistry, natural product organic chemistry, organic synthetic chemistry, and also organic analytical chemistry. Dr. Ambara currently has more than 50 international publications consisted of original article, short communication, and review articles. He also became a principal investigator and co-investigator for several research grants. In this morning, Dr. Ambara will present the presentation titled Reactivity of Acrolein Released from Cancer Cells. Okay, please take your time, Dr. Ambara, for 30 minutes. Time is yours.
Okay, thank you very much. Good morning, everyone. So thank you for the organizer for inviting me to this symposium. It's my great pleasure to join all the meeting here. And also, actually, this is my first formal interactions with LIPI, so I'm very happy to be here. So first of all, let me share. Okay, so I would like to present about the reactivity of acrolein released from cancer cells. So, okay. So even though the moderator already introduced my background, so I just want to mention a little bit about my research background. So until 2020, I worked in Weekend. So it's similar like a LIPI in Indonesia. Actually, it's the largest research institute in Japan. And actually, during my
Sorry, please unmute your voice. Sorry, Dr. Ambara. Please unmute your voice.
Okay, I'm sorry. My apologies. So let me start. Okay. So good morning, everyone. I apologize for the technical problem. So first of all, I would like to thank the organizer and the moderators for the kind invitations. It's my great pleasure to be here. Actually, this is my first interaction with LIPI, a formal interaction with LIPI, and I'm very happy to be here. And so today I would like to introduce my recent work on the reactivity of acrolein, which is released from the cancer cells. So although the moderator already introduced about my background, so I just want to mention a little bit about my previous work. So from 2012 until 2000, I worked at Rikeng. So Rikeng is actually similar to LIPI in Indonesia. It's the largest research institute in Japan. So actually, there's a very good facilities for the chemists. Actually, for example, in Rikeng, we have a large NMR facilities. If you can see here, in one cube here, we have about 10 NMRs. So if you see these 10 cubes and this large room, we have about 125 NMR, which can be used by the researchers. And also, start from this April, I start to work in Tokyo Institute of Technology as an assistant professor. So during my graduate study in Osaka University, I work to isolate and synthesize this kind of peptidoglycan fragment from the bacterial spermaton. So by training, actually, I'm have an interest to do the interdisciplinary research. So since my background is the synthetic chemistry and also the natural product chemistry, I started to think, why don't I do the bio-related chemistry and bioorganic chemistry. And then when I start to work on this area, I also found that my research actually can be used together with the medical doctor for the diagnostic and therapy. So today I would like to talk about this diagnostic and therapy based on our thesis chemistry. So to begin my talk, I would like to show about the breast cancers. So you see that breast cancer is the most common cancer in women. Around 1.3 million women worldwide affected by the breast cancers. Also in Japan and also in many advanced countries, actually 1 in every 11 women affected by breast cancer at certain point during their lifetime. So if you talk about the breast cancers, there are several types of breast cancers. For example, but the most common one is DCIS, the ductal carcinoma in situ, and IDC, the invasive ductal carcinoma. So the difference between these two is that if we compare to the normal breast gland, so this is the milk duct located at these positions. So if you see the normal breast glands, we could see the inside like this. But if you see the ductal carcinoma in situ, you have the cancer cells around inside the duct like this. But in the case of invasive ductal carcinoma, the cancer cells is already spread outside and affected other area. So the IDC is more difficult to treat rather than the DCIS. So there are several ways to treat the cancer, including the radiation therapy, chemotherapy, hormone therapy, and also the surgery. In the case of surgery, there are 2 options to perform the surgery. That is the breast conserving surgery or the breast removal surgery. So in the case of breast conserving surgery, during the surgery, only the affected area removed. On the other hand, on the breast removal surgery, even though only small amount of cancer appear, but the whole part of the breast will be removed. So of course, this breast removal surgery is difficult for the patients because they will lose part of their bodies. But also to perform the breast conserving surgery is also difficult because it's sometimes difficult for the doctor to determine which is the normal tissue and which is the cancer tissues. So if during the surgery some tissue is still remaining, then it will become large again, and it will become the big problem in the future. So during the breast conserving surgery, usually the surgeon will remove some affected areas, and then he will cut this removed area into 4 pieces, and then give this to the pathologist to be analyzed. So the pathologist will analyze these tissues by using the HE analysis. It's a very conservative analysis. So by looking at the morphology of the tissues, these pathologists will determine whether it's normal cells or whether it's cancer cells. And then if it still contains the cancer cells, then the pathologist will talk to the surgeon and tell them to perform again the surgery. So these cycles perform 2 or 3 times, and one cycles need about 40 minutes. So if you can imagine if this cycle needs to be performed 2 or 3 times, maybe it takes 2 or 3 hours. And during that time, the patient is still stay in the operation room with the surgery cut still open. So it's very long process and very difficult process. Also, the pathologist is not always available in every hospital. It's only available in big hospitals. So To overcome this problem, many scientists are trying to develop a new method. For example, this margin probe device. This margin probe device could detect the electromagnetic wave of cancers. However, it's 53% of false positive rate. So sometimes it's not cancer, but it's just detected as a cancer. So it's still not a good method to detect the cancers. And also we know that the micro CT or the MRI also could be available to detect the cancer. But this method detect the presence of cancers and normals, but cannot distinguish the type of the cancers. Also, the chemists also have developed a chemical probe. For example, some groups in Tokyo University has developed this probe, which when they add this probe to this breast cancer stump, only they will react with the enzyme which present on the surface of the tissues, and then release this molecule to give the colors. But the problem is they could not define the difference between each cancer types. I should mention that to differentiate between each type of cancer is important on the breast cancer surgery. So recently, we found that the phenyl azide could react with the acrolein through these click reactions to give this triazolene, which the double bond isomerize into these positions, and also the oxidized product. So the reactions is selective, and it's also stable in physiological conditions, and the azide also it could be performed in this physiological condition, and the azide itself is quite stable. So to show you in more detail, so the azide only react with the acrolein, but with the same compound which has substituent at the beta positions, alpha positions, or the other analog, the azide does not react under the similar conditions. That means that it's only reacts specifically with the acrolein. Also, it could react with the acrolein to produce this compound, which could easily internalize into so that we could observe the fluorescence at the end of the experiment. So by looking at the difference of the fluorescent intensity, we could distinguish by using our probe whether the cells contain acrolein or the cells does not contain the acroleins. So speaking about the cancer cells, actually the cancer also has a high level of reactive oxygen species. This high level of reactive oxygen species could induce the lipid peroxidations at the surface of the cancer. So it will produce various type of aldehyde. So we could find on the literature that this type of aldehyde has been reported to be produced on the cancer cells. However, in the case of acrolein, many publications and review articles says that the acrolein actually presents in the cancer cells, but actually no experimental data for this. So by using our probe, we believe that we could give the experimental data that the acrolein actually induced produced in the cancer cell in high level. So we add our probe into various type of normal cells and also into various type of cancer cells, and we, after several processes, we could just distinguish them by the fluorescent intensity. So if you look at the normal cells, the fluorescence intensity is very low, but in the case of cancer cells, after the reaction with the acrolein, the fluorescent intensity is, you can see that the intensity on the fluorescent intensity on the cancer cell is higher. So to make it easier to be compared, so I made the graph here. So on the left side, this is the normal cells, On the right side, this is the human cancer cells. So the higher the fluorescent intensity means the higher the acrolein level on the cells. So you can see in the various type of cells, the various type of cancer cells, the acrolein level is higher compared to the normal cells. Also, these on the right side, all of this type is the breast cancer cells, and they have a higher level of acrolein compared to the normal cells. And also, at this point, we use the TAMRA as the fluorescence group here. And we found that this molecule is the best molecules for our probe, because when we just change the fluorophore into different type of groups, for example, like coumarin, Janelia fluor, or cyanin, it could not give the best performance. So this TAMRA phenylazide is the best combinations for our probe. So we determine the mechanism how this probe works. So in the cancer cells, there are a lot of acroleins produced. So when we add our probe into the cells, the probe will react with the acrolein from the cells to produce the triazolene, and it will give the diazo compound. So because the low intracellular pH conditions, it will easily to produce a diazo compound so that it could immediately react. So this diazo compound is very reactive. It could produce carbon immediately, and so that could react immediately with the nearest organelle. So they make the covalent bond. So that's the reason why we could visualize the cells, visualize the fluorescence on the cells. And this is, we make more details, we observe in more details that the fluorescence, that the probe when go inside the cells, when they not react with the acrolein, will just go outside, but when they meet the acrolein, they will react and they just attach covalently with the organelle. And so we know the mechanism, and we observe that it's specific with the acrolein from the cancer cells. So we just, what we think that maybe our probe also will work with the real tissues from the cancer. So we worked with the breast cancer surgeon. So they are one of the best cancer surgeons in Japan. They're from the Osaka University Hospital, and they gave us the real sample from the breast cancer patients. And when we add our probe to these samples, and after wash with the buffer and observe under fluorescence, we found that we have a lot of picture actually, but I will show you only the representative data. So in the case of normal tissues, we only found the blue colors, which come from the nuclei that visualized by the Hickes reagent. But in the case of the cancer tissues, we found the red fluorophore, which come from the acrolein that react from that visualized by our probe. So we collect all the data and make the statistic on this graph here. In the case of normal tissues, the intensity is very low, but in the case of cancer tissues, the intensity is quite high. So in this case, we could observe the difference between the normal and the cancer tissues. But this is not enough, because we need to differentiate these two types of cancer tissues. So we just observe in more detail their morphologies. So under the 200 times of magnifications, we could see that the morphologies can be observed by our probe. So you can see that these normal cells and these three types of cancer tissues cells has a different morphologies. So we can differentiate these type of tissues. And when we compare to these classical H&E analysis method, they give the similar result. The difference is that this method needs about 40 minutes to be performed, but our method only only needs about 5 minutes to be performed. And also we ask the pathologists to perform the analysis. We ask them to do the blind analysis and compare our method with the classical method, and they found that the result is quite similar. So to conclude the first part, so I should mention one more time that by only looking at the fluorescent intensity, the normal tissues can be differentiated by the fluorescent intensity with the cancer tissue. But when we want to differentiate these two types of cancer tissues and these two types of normal tissue, we need to see the morphologies. And only so far, only our method, our probe, which can be used to visualize the morphologies other than the classical method. But our method can be performed only within 5 minutes. So this is the molecules that we showed on the previous slides. It's the first generations. We use on about 20 micromolars of the tissue of the concentrations, and we now have the second generations by adding this isopropyl group, the bulky group here. The intensity can be increased, so we only need 2 micromolars. And this method has been used in the hospital in Osaka University, and it will be used in all over Japan in the near futures. And we just did the press release conference at the Mambuka's office, and I think it will be soon. We want to use it also in outside Japan. So that's the result for the synthetic. I named this method as the synthetic chemodiagnosis, and also the rest, the remaining time I will talk about this synthetic chemotherapy. So previously I mentioned that by adding this isopropyl group here, the bulky group, this azide, it has a higher reactivity compared to this simple azide. So we just, to prove it experimentally, we just mix this simple azide and this bulky azide and react them with the excess amount of the acrolein. And we found that only this bulky azide could react with the acrolein, but we could not find the product from this simple azide. It means that this bulky azide has the higher reactivity compared to this simple azide. To explain this phenomena, we performed the DFT calculations by performing DFT calculation with our collaborators in Hokkaido University. And the reason is because the transition states of the reactions between the bulky azide and acrolein is lower compared to the transition states of the reactions of simple azides with the acroleins. So I will not talk in more detail about, so this is more detailed information about the calculation method, but I will not talk too much detail on this part. Just to conclude everything is that the bulky azides actually is more reactive compared to the simple azides. Also, when we look into more detail, interestingly, this bulky azide could react with the acrolein to give this triazoline, and it's react further to give this triazole compound and also this diazole compound. This diazole compound is quite stable because it's located at the alpha positions of the carbonyl. Even we could isolate this compound, and also previous report also mention about this alpha diazole carbonyl compound. So this is actually interesting phenomena because when I think we could use this compound as a therapeutic method, which I will show you on the next slides. So if I design this compound, the drug with the carbamate link to this bulky azide, usually some drugs have the amine group, which when it's alkylated, the toxicity is reduced. So this compound, if they just add it into the normal cells, they will not show any cytotoxicity. Was added to the cancer cells, which has a lot of acrolein, they will react to produce this diazo compound. And if you see these lone pair electrons, it will push the conjugate bond to release the drugs and give the active drugs only at the specific cancer cells area, or cancer area. So it will kill the cancers here. So we would like to do these reactions not only on the cells, but on the xenograft mouse. So our laboratory has the ability to prepare this xenograft mouse. So we would like to just prepare the cancer on the mouse. So it means that they have a lot of acrolein now on the cancer area. And when we add our prodrug, the reactions will, they will react with the acrolein on the cancers to give this diazo compound. So as you can see here, after 4 hours of reactions, the coumarin starts to be released and we could observe the fluorescence here. But when we just add the glutathione, the azide does not react and it's not reduced. The starting material still remain, and that our azide only reacts specifically with the acrolein. So we just add our probe, our prodrug here to the cancer cells and also to the normal cells. If they react with the acrolein, the coumarin will be released and we could see the increase of the fluorescent intensity. And the result is shown here. When we add to the normal cells, the fluorescent intensity does not increase, means that no reaction occur. But when we add to the cancer cells, you can see that the fluorescent intensity gradually increase, which means that the coumarin gradually release after the reactions with the acrolein. So we just changed now the fluorescence with the drug. We use the mitomycin C. So this drug, when we alkylate it at these positions, they shows no toxicity. But after the reaction with the acrolein, the drug will be released to show the cytotoxicity. So at the cytotoxicity test, we found that this mitomycin could have the cytotoxicity, non-selective cytotoxicity against normal cells and also the cancer cells. But when we perform the cytotoxicity test on our prodrug, the normal cells doesn't show any cytotoxicity. But the cancer cells, since it has a lot of acroleins, our prodrug react with the acrolein and they release the free drugs to kill the cancer cells. So now we are performing our experiment on the real, on the cancer xenograft mouse. So I just show you the result here. So we just add our prodrug into the cancers. If you see this is the control experiment. If the azide does not have any drug here, after 14 days, the tumor is increasing. When we add mitomycin, the tumor is gone. But you can see that the inflammations occur everywhere on the mouse. It means that the adverse drug reactions, the off-target is occur here. So it's not selective. And this mouse is just, we have to sacrifice, the mouse is dead after about 18 days. But when we add our prodrugs, we can see that after 14 days, the cancer is, we can suppress the tumor growth. And also the mouse could survive even more than 50 days. In this case, we injected our prodrug directly to the tumor. Also, in a different experiment, we injected our drug intravenously. You can see that in this control experiment, after 10 days, the tumor cannot be suppressed. tumor can be suppressed, but the inflammation occur. So it is very toxic to the mouse. But on our prodrug, the tumor can be suppressed and also no inflammations, and the survival rates also high for our product. You can see also the tumor growth here and at this graph. So to conclude my talk, so we use just the simple reactions of the azide in acrolein, and we apply this into the synthetic chemodiagnosis for the applications in diagnosis of the breast cancer patients. And we also have performed this synthetic chemotherapy for the therapy of cancers. Now on the animal levels, we have performed with the good results. And finally, I would like to introduce our laboratory team. So currently I'm the assistant professors of the Professor Katsunari Tanaka laboratory, and he has given me a lot of freedoms to do the work. And now I work with these 3 students, and also this one lady, Penny Sang, will go to LIPI, I think this next year, and he will start the new work there. And also I'm very grateful for the Kakenhi, the very generous research grant from the Japanese government. So that's all of my talk, and I would like to thanks for your kind attentions. Thank you very much.
Okay, thank you very much for nice and important presentation, Dr. Ambara. So we still have one more keynote speaker, Professor Chaudhary from Pakistan. But Professor Chaudhary will be able to join with us at 10:30. So now still 10. So we have about 30 minutes before Professor Chaudhary join. We will give opportunity again to Professor Gunawan to complete his presentation. Yeah, as we know, the last part of his presentation delivered so fast. So please, Dr. Gunawan, keep slow to explain important of your result. So maybe if we still have time before Professor Chaudhary join with us, maybe we presenter will ask will answer some question if we have a time. Okay, please, Professor Gunawan, deliver and complete your presentation. Hello. Okay, please unmute.
Hello, can you hear?
Yeah, okay.
Yeah, thank you.
Okay.
Thank you for your time. I will share first the screen, the slide.
Okay.
Can you see the slide already?
Not yet. Maybe need.
Can you see the slide?
Yes, I can.
Okay. Okay.
Residual toxic metabolite for determination, they replace this one, use by 10. This is how to calculate the detector's limit of the standard, pure standard. But if we use sample, any sample, leaves, root, or etc., we should determine the sample detection limit according to this regulation of FDA. For this purpose, we should spike the matrix with the concentration of limit of quantification with 7 replication, and then determine the content and determine the standard deviation. By this method, the detection limit can be calculated with this equation, and the quantification limit can be calculated with this equation. So it's mean for every matrix, it could be the detection limit is different. So that is why the detection limit must be performed for each of matrix. The detection limit in blood may be different from urine or maybe different from other metric. So if we work with leaves and we have work with root, so maybe the detection limit is different. So it is important to determine what they call sample detection limit. After we get sample detection limit, we should verify or confirm the data by determining what they call cutoff value. This cutoff value can be calculated by using this curve. This is the concentration, and this is the positive result of the detection. For the example, this is the detection limit is about 80 microgram per liter, and the maximum permitted level is about 125 microgram per liter. So by doing experiment, it is nice 10 times each of concentration, and we can calculate the positive result, percent of positive result. So the cutoff value can be calculated by the value if the false negative is below 5%, something like this. So it is possible that the detection limit is should be below cutoff value, and the cutoff value should be below the maximum permitted level. This is the guidance of Eurochem Guide 2014. And then I continue. ISAC can be defined as analysis of a drug to quantify the biological activity or activity of one or more components by determining its capacity for producing an expected biological activity on a culture of living cell in vitro or a test organism in vivo. That is the definition of the USP 43. Maybe other guardians have a different definition, but the aim of performing biological assay or bioassay is to determine the effect of a substance on a certain type of living matter or a biological test system. For doing that, some steps should be taken. First, we should do literature survey for selecting the correct bioassay. I mean, what we have the equipment, what we have the cells, so we should survey from the literature and then select. After that, we should evaluating the stability of the extract or compound on the selected solvent. And then after that, we should testing the performance of the biosystem using positive and negative standard. Also the same thing with, we should first authenticate it. And stability of the test sample, extract or extracted compound and reference standard must be evaluated in the selective solvent. I recommend using chromatography. If it's not stable, so something must be done. We cannot process if the sample is not stable. So this is some of method of authentication of organic cell by using ATCC method. You can download this method. Selecting the organism, cell line, or bioassay should be based on the used formal guidance. For the example, WSO or other guidance as references. This is some example. For leishmaniasis, we shoot this cell line. This is a number of ATCC. So on, we can have this guide from the WSO, etc. There should be certain cell line. The biosetting should be selected according to the news guidance or reference. Analogy also for the reference standard. So usually we will get something like curve, sigma-like curve. If we do some bioassay, that is the response, that is the concentration, that is maximum, minimum, that is the IC50. From this, we can calculate what we call Z factor. The evaluation of biosystem sample performance can be authentic standard and sample, then calculating the Z factor. Is standard or sample? That is the equation. That is the maximum. That is the minimum. Bottom minimum. That is the calculation for Z factor. That is a standard deviation of positive control, negative control. This is the average positive control, average data, and this is average negative of negative control. This is the equation, the same equation, but this is the requirement. So it means if we do something using certain cell line, what we first do is using the suitable standard to determine the Z factor. The Z factor must be more than 0.5 or at least it's not negative. That is the requirement. If the Z factor is not acceptable, it means that the system is not suitable. After the system is suitable for the standard, the same thing we should do with our sample. The sample should also give acceptable Z factor value. This is very important because without this method, we cannot show whether our Bayes system is okay or not, right? Our Bayes system is okay or not for our purposes. So that's why we should try to evaluate this Bayes system, I mean the cell, the standard, etc., using this equation. After that, we continue to determine what they call selective index. Selective index can be defined as the ratio of the toxic concentration of a sample against its effective bioactive concentration. The ideal drug should have a relative high toxic concentration, but with a very low active concentration. This is the equation. This is IC50 for cancer. This is a cancer cell, no cancer cell. This is for cancer cell. SI should be as high as possible. 10 is nice, but below 1 is not acceptable. If the value 1 to 10, we should try other experiment, other Bayes system to confirm the result. If the selective index is too low, it means the compound or the extract is too toxic. I think it's no need to further investigate it because it is too toxic. For continuing the work for in vivo or clinical study, the accurate potency of the tested material must be determined. This is needed to decide the correct administration dose if we want to do animal evaluation or clinical evaluation. For this purpose, the author, I recommend you to apply the method of European Pharmacopoeia 10/2020 section 5.3. This section gives you complete equation. We can just set the equation into Excel and then we can do manually with complete with many parameter or many conditions. Or we can use also from USB 43, but this the equation is not complete. They recommend using computer, but I recommend this using European Pharmacopoeia. That is nice. We can do it manually by Excel only. This is the conclusion and recommendation. Indonesia is very rich of traditional herbs and other natural resources that can be developed into phytopharmaceutical and or modern drug. It is recommended to determine the quality marker and its specification range, I mean the maximum and minimum content of each of the herbal monograph that described in Pharmacopoeia Herbal. Performing clinical trial for all marketed phytopharmaceuticals is needed to have a consistent quality of every batch of the product. If we have consistent quality of the product, I think the medical doctor will very happy to apply this herbal drugs. For having reliable conclusion, all use chemical and biological assay must be first validated according to the use guidance before it can be applied. If the use method were not first validated, the conclusion could be wrong. Due to the limited funding and facility in Indonesia, collaboration between scientists are very important to develop the phytopharmaceutical or modern drug from Indonesian herbs. Coordination between university and research institutes are very crucial to prevent the duplication of research, and the limited funding can be efficiently used. I think if we can do collaboration from all university in Indonesia and LIPI to be the coordinate all the research, I think will be nice. This is, I will thank to Dr. Abdi for the nice invitation. And once again, thank you for your nice attention. It will be very nice if we can do collaboration. Do you agree, dear distinguished participants? Thank you. Good day. Nice to meet you all.
Okay, thank you very much, Professor Gunawan. Now we still have about 15 minutes before Professor Chaudhary join with us. So maybe we will have a little discussion. Yeah, audience already raised her hands. One audience, Ibu Emma Damayanti, but we don't know from which institution. She want to deliver her question to Professor Gunawan directly. Maybe organizing committee can unmute Ibu Emma Damayanti and answer about this question.
What I'm talking about is how to do a complete validation method or how to do quality control. This will be done if we have equipment. We need many equipment to do that. But if it's not, I think we can do very simple. We can do by the public jamu or the home industry can make a note at first with very simple to note all of things regarding the drugs, where they got the drug, where the place, and the age and the part of the plants, and then to note also how to make the drug, and then they can ask the consumer how about the drug, is this okay or not? By doing this, someday if we have complete data, we can conclude it if the plant is collected in a certain places, they will not active. Or if the plant collected with certain is, that is not active. That is very simple, but we need to do some survey and we do a work by many people, and we can collect together all the data, and we can do something metabolic profiling, but without any chemical method, without any, but just be asking the people, the consumer, about the drug that they can produce, because the drug may be produced with different way. This is my question. This is nice if someone do this survey, all of the survey, and then at last we have conclusion. This plant is active if cultivated on a certain place. This plant is active if the harvest on certain months or something like that. So the plant is active is the mode of extraction, something like this. At last we will have something like that. And but if we have chemical equipment, that's the same. We will do profiling, chemical profiling. We will do IOC profiling. Regarding the patent, maybe the participant know better than me. I do not know about regarding who will get the patent of the research. I think both of them who work with this work will get the patent. That's my answer. Maybe it's okay.
Okay. Thank you, Prof. Okay, thank you, Prof. Gunawan. maybe before conduct the research, if jamu producer and researcher would like to have joint research, maybe it's Professor. How do you think in Indonesia is the regulation body like BPOM applied the rule of marker of herbal medicine in standardized traditional drugs or phytopharmaca? Okay, please.
Okay, thank you
Professor
for the nice question. As I described previously, Pharmacopoeia Herbal Indonesian or USP 43 or European Pharmacopoeia, they just only describe the minimum standard, the minimum concentration of certain compound or a group of compound. This is generally something like that. The maximum compound, the maximum concentration was not described. This is that's why by this reason Professor Ringer wrote in his publication that it's not enough for ensuring the quality of the drug, because if it's just determining the polyphenol or flavonoid or certain compound, it could be not correlated with the activity. The drug will be okay if they can be used by the customer. So that's why it is need to determine the quality marker or marker ranking system 3 to 5 based from the plan, the actually quality marker. Unfortunately, in Pharmacopoeia Herbal Indonesia and other present Pharmacopoeia USD, this was not described. This is only described by some publication, but just only a few. That's why if we do quality control using any marker, maybe it is okay, but the pharmacology action is not the same batch to batch. This is the problem. That's why it is nice if we start from Indonesian medicinal plant one by one to search what is actually the quality marker. This is not yet described in any pharmacopoeia of the world, current pharmacopoeia.
Okay, thank you, Professor Gunan. Maybe
if you must have another comment of my answer, let me know.
Yes, Professor
Okay.
Gunawan, I can accept your idea. Yes, I think like that in Indonesia, we the BPOM cannot apply all of the rule of market that you say in your presentation.
So that's why it's nice if LIPI or PALEPOM coordinate the research in all Indonesia's one by one. So there is application, there is no efficient. They can think that's nice. That's my dream.
Yes.
40 years ago.
Yeah. Nice, Professor Gunawan. Nice. Thank you very much, Professor Gunawan.
Okay, thank you, Ibu Marisa. Yeah, maybe we move to another question. Now, question for Dr. Ambara from Pak Ahmad Sarifuddin from LIPI. The question is, as we know that the cure treatment for cancer is
need to be treated in a specific way. So also the method, there are always the advantage and the disadvantage. So in my case, I focus on the first part of my talk. I focus on the method to support the surgeon during the diagnosis of during the cancer surgery. And on the second part, we use this product to eliminate the off-target reactions. So actually, it depends on the stage whether we can do the non-invasive. But actually, even though we perform the non-invasive, it doesn't guarantee that in the future the cancer will not regenerate again. So as far, I think so far also in the advanced country also, there's the way the method is still very, could be different for each person. That's even though in Japan, as far as I know. Okay.
Okay, maybe it's clear. There is more question to Dr. Ambara from Ibu Sylvia Lekatompesi. Yeah, the question is, my cousins, young person use traditional medicine, turmeric and suchi leaves. Cancer does not invade other organs, and cancer cell out of the breast. But now she knew to the hospital. My question: can the patient use your treatment and survive? I hope this result can help women who are sick with cancer, can be useful for overcoming cancer. Okay, can you explain, Dr. Ambara, please?
When we start to start our research, our purpose is that we would like to make a contribution for the cancer treatment. So at the first part of my talk, I mentioned about the chemical probes that has been used in the hospital in Japan. It's easy to be approved. The method is easy to be approved because on that method, the cancer was taken out from the patient bodies. So it was taken outside from the cancer, from the patient bodies, and then we treat the stump by our probe. So we did not put any reagent into the human. So that's why it's easy to be approved, and now it's already started in the Osaka University Hospital. But on the second part, on the chemical, on the prodrug that I have been taught on the second part, it needs to be injected into the patients in the future if it's approved. So there's still a lot of several steps to be tested, but on the animal levels, it shows a very promising result. So we are trying hard to make it fast to be applied further in the near future. So hopefully we could contribute in this area also. Okay.
Okay. Ibu Sylvia also asked her cousin use a traditional medicine, turmeric and suchi leaf. Do you have comment for this part?
So actually, I'm not the expert for this traditional medicine. Professor Gunawan maybe can answer this question, but we are focusing on the drug, on the pure drug that can be used can be linked to our prodrug. So I cannot comment on these traditional medicines.
Okay, it's okay. Okay, already 10:30. Ibu Marisa, Professor Choudhury is already joined with us or not yet?
I think not.
Oh, Pak Abdi, maybe no.
Maybe we can extend for discussion around 15 minutes more.
Okay.
Because
Prof. Iqbal has not joined us.
Okay.
The breast cancer surgeon, when we published the paper, he tested on several stages of cancer, even stage 1 to stage 4 of cancer, and our probe could be accurately detect the cancer for this kind of breast cancer. So that could be used to guide the surgeon until which area need to be removed, so that the reoccurrence can be we can just the recurrence hopefully will not occur again in the future. So we tried with from the phase 1 to phase 4 of the breast cancer, and now also more data has been collected, but for the details I could not say more about the result for this point. But on our previous publications, we tested on the phase 1 to phase 4 of the cancer. patients.
Okay. It means it's possible to use in early stage.
Yeah, hopefully, yes.
Yeah. Okay, maybe it's already clear. Okay, we move to next question. Come from Ibu Teni Ernawati from LIPI also. The question is, it is nice presentation and also full of knowledge, Dr. Ambara. From the results of the research you have done, are there always significant differences for cancer cells and normal cells against the anti-cancer chemical compounds that have been synthesized?
Okay.
Okay.
So first of all, I think this is Buteni from which come from to Osaka several years ago, if I'm not mistaken.
Yeah, probably yes.
So yeah, I mean, we tested to several types of normal and cancer cells with our products, and they give almost the similar results. So we could see the difference between the normals and the cancer cells. So far, I show you only one drugs in this experiment and these presentations, but actually we tried several more drugs, which could not be shown now in the, today because we still need to publish and patent the data. And everything seems good. They give the clear difference between the normal cells and the cancer cells. I'm sorry I could not show the data for now, but hopefully on the different opportunities I could show you in more details after we publish the results in the near futures. Thank you.
Professor Gunawan, about the quality marker, how we can get the reliable data of the marker. Is it possible if we only examine only one tool, such as TLC scanner or densitometer? Thank you. Please, Professor Gunawan. Okay, sorry, your voice is still mute.
Okay. Sorry. Thank you for the nice question. I think for determining the quality marker, it should do some chemical profiling and some ISAC profiling. For chemical profiling, we should have LC-MS. That's impossible if we just only using TLC. If we have TLC, and then we should isolate the fraction of the fraction of the spot and determine the structure. That is my answer. But if we have LC-MS, we can directly predict or determine the quality marker. If only using TLC, we can do the same thing, the TLC profiling and ISAC profiling, and by combine with using multivariate, we can predict which spot that active, which spot that is not active. The active spot can be isolated and the structure can be determined. That's my comment. Maybe.
Okay, thank you, Prof, for the answer. It's very useful for us. Thank you very much.
Okay, thank you. I already saw Professor Chaudhary already joined with us. Welcome, Professor Chaudhary. Maybe your voice is still mute.
Assalamualaikum warahmatullah. Good morning.
Warahmatullahi wabarakatuh. Welcome to our position. Okay. Are you ready to present your presentation?
Yes, inshallah, whenever you want me to.
Okay. I will introduce our third keynote speaker, Professor Iqbal Chaudhary. Professor Chaudhary is a distinguished national meritorious director at the International Center for Chemical and Biological Science, HEJ Research Institute for Chemistry, and Dr. Panjwani Center for Molecular Medicine and Drug Research, and Coordinator General COMSTEX, Organization of Islamic Cooperation Standing Committee on Scientific and Technological Cooperation. He has written and edited 76 books, most of which have been published in US and Europe. He is also the author of the over 1,000 research paper, wow, in top international science journal of the West, as well as 55 US patents. He has been among the most cited scientists in Pakistan in last 5 years, with citation exceeding 25,000 citation, with H index 68. Professor Chaudhary has skillfully used his deep understanding of chemical principle and biological process in the discovery of a large number of fascinating molecules with a potential therapeutic application. In this morning, Professor Chaudhary will present the presentation titled Integrated Natural and Synthetic Chemistry-Based Approach in Heat to Lead in Drug Discovery, example of our recent work. Okay, please, Professor Chaudhary, time is yours.
Assalamu alaikum. Good morning, ladies and gentlemen. Thank you very much, Professor Andrea Augusta, for a very generous introduction. It's always a pleasure to speak to our brothers and sisters in Indonesia. Indonesia is my second home, and it's always a pleasure to be with you. So I'm absolutely delighted to be part of this interesting conference, though I really wanted to visit you physically, but hopefully, inshallah, soon when situation will improve, we would be able to have this event physically. So what I would do is to share with you my slides. Let me take you through slide share, please. Okay, so next 45 minutes, I would be speaking to you. About this interesting work which we have been carrying out since last several years, and that is the use of natural product and synthetic chemicals in the discovery of medicine. And as we have mentioned during my introduction, that we have got many patents from different countries, and we have developed many lead molecules which are in different phases of development. I'll just give you an example of a few. It's very important. You work in LIPI also. Many places in the developing world, we have a huge reservoir of natural products, and we have been using them for a variety of different purposes. And these are examples of some natural products which have made to the world scene as drugs. So whether it is quinine or morphine or Taxol, all of them are extremely important, and they have been discovered out of ethnobotanical practices and the science mixed with it. In Pakistan, we have a long country not as big as yours, but this is a one single piece of land, about 2,700 kilometers long from this corner to this corner, an extremely diverse country, because from tropical shores to deserts and subtropical areas to Siberian climate, all mountains and lots of snow. We have very diverse flora, and that diverse flora allow us to work on very large number of nature products. And in the process, we have collected over 5,000 plants. We collected those plants along with the information of their use in different cultures for therapeutic purposes. And then out of them, we have conducted lots of different studies. So primarily information about their folk use, and then by using scientific methods, bioassays and phytochemistry, we were able to isolate very large number of compounds. I'll just give you example of a few which made it to the US patent, and they are in different phases of development. ISOX, which was isolated. ISOX is the name of a drug candidate which is in phase 2 clinical trial. This was isolated from Delphinium dernitatum, and it is one of the strongest anti-epileptic natural products. Then there is a health supplement which we have developed for Parkinson's disease. This is also recently patented in USA. We have also developed topical formulation for various skin diseases, including leishmaniasis, and that is also patented. I'm going to present to you briefly about 3 projects, and these 3 projects based on very large number of natural products and synthetic compound which we have in our library. So we have a large bank of diverse natural and synthetic compounds, and these compounds have very interesting structures, and these structures are primarily the basis of their function. But before that, I'll just show you what new skeleton we have introduced into the world literature. World literature is now full of our discoveries. You would find from one single group, as much as we have published, no other research group in the world has introduced so many chemical compounds in the field of natural product chemistry. For instance, this is a triterpene, which we have isolated in fascinating cage-like structure, which we have a compound with N oxide isolated from Nigella sativa. This is a diterpene which was isolated from a marine from marine source. As you can see that there are lots of interesting skeleton, because these are secondary metabolites, and it gives a fascinating structures which may have very interesting biological activities. So anthamine, a very interesting steroidal molecule, a series of compound which was isolated from a marine plant, and of course lots of other interesting molecules, but just wanted to give you the glimpses of absolutely interesting structures which you can expect from natural products, because nature provides you molecular inspiration that gives you interesting skeleton which you can overbuild. Now, this is the first project which I wanted to discuss with you briefly, and that is related to resistance in microorganisms. We all understand that bacteria, fungi, and parasites are developing resistance, and these are bad bugs. You know, for instance, it's called ESCAPE, in which we have 6 of these microorganisms declared as some of the most dangerous microorganisms in the world, and they have developed literally resistance against all known antibiotics. I'll give you an example of our work, though this is a big project and I can speak about that for several hours, but I'll just tell you how important it is to work in this particular field. Resistance is everywhere, and we all are affected from microbial resistance. When we get sick, it becomes very difficult for physicians to treat us because microorganisms are often resistant against the available antibiotics. Now, resistance is a cross-cutting, is everywhere from viruses to bacteria to parasites, and there are lots of different reasons, but this is a globally alarming situation. Now there are three different options in which one can work. You can discover new antibiotics. That takes lots of money, about $2 billion, about 10 years, and you never know how long this antibiotic would continue to function, because resistance is often emerging within a decade. The second option is structural modification of existing antibacterial agents. This is also an interesting approach, that you take existing antibiotics, you modify their structures, and that you hope that this would work for a while. And that is also a good approach. And many of the antibiotics which you see in the marketplace are the modified form of existing antibacterial, antimicrobial agents. But what we have done in our institute, in our research group, is to actually use agents which can modify and modulate resistance in antimicrobial in microorganisms. Primarily, the objective is that you have antibiotic and then you have a modulator which help the antibiotics to work. So these are called resistance modifier. These are so-called resistance reversal agents. And I'll just give you an example of, though we have isolated a lot of them, many of them got patented internationally also, but this is an example of a natural product isolated from a Morus species. And you can see this is primarily a flavonoid, and this is loaded with lots of functionalities. Here you can see that this is 91% active against AMRSA, which is epidemic methicillin-resistant Staphylococcus aureus 17 species, and you can see that this is active more than the vancomycin. So this is a molecule which was found to be more active than the drug of choice, vancomycin. This is another molecule which was isolated from the same plant, and the only difference between the previous one and this one is the presence of this isopropyl unit, but the activity has reduced. I'll just take you what we have done. So we have selected this one compound, and we have carried out lots of morphological studies by using microscopic methods, and we have done lots of molecular studies by using flow cytometry. And by combination of these methods, we were able to have some idea of the activity of this molecule, which is called arginine. This is Staphylococcus aureus, which we have targeted. We all understand Staph aureus is an extremely important bug, is gram-positive. It is nosocomial and is also community-acquired, is a normal flora of human body. And but that becomes pathogenic because a lot of systemic and lots of localized infections are caused by Staphylococcus aureus. This is a picture of a normal colony of Staphylococcus aureus, and when you expose this colony with this particular compound, arginine, then you see a very interesting structure. You see that the colony is simplified, not many bacteria survived, and even the one which survived, they have differing morphology. So morphological changes were noticed through microscopy, and by understanding, for understanding the effect of it, we have carried out a very interesting study in which we have used propedium iodide and compound and antibiotic. And as you can see that without the compound, without this compound, the number of cells which accept propedium iodide are very few. But when it is in combination of compound and antibiotic, then you can see this red region, which means that larger number of microorganisms have accepted the dye, and which actually indicate that this compound is capable of helping antibiotic to enter into the cell And this is scanning electron microscope, in which you can see the grape-like structure of Staphylococcus aureus. AMRSA is 17, but when it is exposed to this particular compound, a completely different behavior is seen. Morphology changes. You can see the cell division is abnormal, biofilm is broken, and at the same time, you can see that the colonies are simplified and cells are disintegrated. This is an interesting picture, transmission electron microscope, in which you can see the cells nicely dividing in two parts, binary fission in a normal colony. But when it is exposed to arginine A, then the morphological changes are so obvious that you can see the cells are abnormally dividing, cells become hollow, the subcellular organelle come out. But this is a fascinating one technique in which you can look into the cell at the nanoscale. So you can measure the cell height, you can measure the cell dimension, and the color represent the cell height. So on the scale site, you can see by using atomic force microscopy, the cell can be measured at a nanoscale. This is the normal cell. And when this single cell, which is about 27.5 nanometer in height, when it is exposed to arginine, then amazing thing happened. You can see the cell height has collapsed, cell collapsed to 0.75, literally nothing. And you can see most of the organelles in the cells of Staphylococcus aureus come out of it. So complete disintegration of cell is measured, and is an extremely interesting technique in which you can see morphology and measure what happened to the cell in real time, because you don't need to dye them, you don't need to fix them, you can do it in real time. So you can take the picture of cell, and you can look into the behavior of cell in the presence of this compound. Now, why it actually happened is still a mystery. We are still working on it, but one theory on which we have worked and found some indication is that this compound arginine is capable of inducing the production of reactive oxygen species, peroxide. And so this bacterial cell, in the presence of arginine A, produced lots of reactive oxygen species. And as we know, reactive oxygen species are free radicals. They can damage the cell wall, they can damage lipid membrane, and they can also damage DNA. So a whole range of damage was actually observed in this case, and this may be the mechanism of cell destruction. There's some antibiotics which function through the induction of reactive oxygen species. This is an interesting slide in which you can see on the side various antibiotics. So you can see erythromycin, streptomycin, clindamycin, so different antibiotics. And these four bars represent what actually happened. In the absence of, I'll give you an example of clindamycin. In the absence of this molecule, you need to have a large quantity of clindamycin, about 2,000 microgram per ml. But in the presence of this arginine, you need only 8 microgram per ml of clindamycin. So this molecule is capable of helping classical antibiotics to enter into the cell and function. Now we have worked on understanding the chemistry of cell, and this is a protein which was isolated from Staphylococcus aureus resistant strain. So we know that there are certain proteins which are different in susceptible strain and resistant strain, and this protein was expressed, its structure was done by using NMR spectroscopy. And here you can see the structure of a protein which is different between susceptible strain and receptor strain. Similarly, a number of other proteins were isolated, and this study was conducted by using various spectroscopic techniques. There are some of the international patents which we have obtained on resistance reversal agents, not an exclusive list of patents. There are many more. But I'll just, because I still have about 20 minutes, and I'll just want to take you to two more projects. And this is related to, again, a very interesting work which we have conducted in last 6 years or so, 7 years. We understand diabetes is a disease which is affecting lots of people. There is no medicine available for diabetes, and the ultimate aim of anti-diabetic therapy is to produce insulin in enough quantities in our body that is capable of handling the sugar, which is called postprandial sugar level. So achieving normal glucose level in blood is the outcome of it. And there are various types of drugs which are available to handle that. Now, what actually happened in diabetes is something very interesting. You know, we keep using anti-diabetic medicine, but we slowly and gradually start losing pancreatic cells. Now, pancreatic beta cells are extremely important because they are the one which produce insulin. And slowly and gradually with chronic diabetes, the number of cells start decreasing. And as number of cells start decreasing, capacity to induce insulin within the body become hampered, and our body cannot really produce enough quantities of insulin. So beta cell mass and beta cell function is extremely important. And why it is important to protect beta cell mass? Because we need to have insulin produced in enough quantities in response of glucose. Now, there are many drugs available, but of course, ultimately, the actual strategy is to prevent beta cells from apoptosis. Why beta cell die? Because diabetes is unfortunately a pro-inflammatory disease. This is a disease with a chronic inflammation, and in diabetes, there are lots of cytokines which are produced in our bodies, which eventually kill beta cell mass. And that is the model. What we have done was that we have used 3 pro-inflammatory cytokines, and we use beta cell. And by using these pro-inflammatory cytokines, we induce apoptosis, programmed cell death in beta cell. And we wanted to mimic what actually happens in a chronic diabetic condition in a patient. So what actually happens is that many of these cytokines start attacking beta cells and their number decrease through apoptosis. We wanted to develop a very good model. So we have this cell line, which is beta cell line, and we had these 3 cytokines, and we in the absence of these compounds, beta cell die because these cytokines are capable of inducing the cell death. And this can be measured by caspase-3 activity, by measuring ATP level. ATP level is the indication of cell viability. If low ATP level is detected, it means cells are less. Caspase-3 is an enzyme which is responsible of apoptosis. So if there is a high level of apoptosis, you see high level of caspase-3. So there are various molecular markers through which you can see whether your compound is capable of protecting beta cell from apoptosis. Lots of different natural products per screen. I'll give you an example of only one, berginin, which is a very famous natural product. And this has been from a large number of plants. This is a dihydroisocomerine loaded with lots of hydroxychrome isolated from various medicinal plants. Berginin is a very common natural product, and you can get them in any quantity because this is a very common secondary metabolite which can be isolated from various plants. What we have done was that we used the cell model, beta cell of mouse, and then we induce apoptosis by using cytokines, inflammatory markers. and we use burgeoning. So here you can see that in the absence of burgeoning, you see this is the normal cell without cytokine. So you can see that ATP level is high. In the presence of cytokine, the ATP level is very low. It means few cells. And in the presence of compound, burgeoning, the number of cell in a dose-dependent manner, cells are protected, and ATP level is as high as physiological normal cell condition. So here you can see that this compound is capable of protecting cells from cytokine-induced apoptosis. This is another important cellular marker, caspase-3. We understand that caspase-3 is an extremely important enzyme, is an intracellular surrogate of the apoptosis. Caspase-3, a converging point on which many of these apoptotic pathways merge, and caspase-3 is at a higher level in a cell which is going through apoptosis. So what we were able to demonstrate that caspase-3 is very low in normal cells, is very high in cells which are insulted with cytokines, and in a dose-dependent manner, this compound is capable of reducing the caspase-3 in a dose-dependent manner, which means that it is inhibiting the apoptotic process. So this is the kind of studies which were conducted. This is mitochondrial membrane potential. Diabetes is a mitochondrial disease in which mitochondria get sick, and mitochondria has a membrane. And this membrane potential is an indication of the health of mitochondrial function. So this compound was capable of restoring the mitochondrial membrane potential, which is the indication of. But ultimately, I'll just show you what actually happens. What you want to see is primarily that when glucose is provided to beta cell, the cell should be able to produce insulin. And this is an interesting slide, because here you can see that in a dose-dependent manner, dose of a glucose, this compound was able to protect the cell, and as a result of it, the insulin are produced in large quantities. So this is insulin secretion, and that indicate that this particular compound is capable of not only protecting the beta cell mass, but also the function. Function of beta cell is to produce insulin. And here you can see insulin produced in response of glucose, which is a gold test. This is a very interesting topic, still not fully explored. And a lot of studies are being conducted now in the world, because this is a new line in which one can develop anti-diabetic medicine. The last project which I would present to you is for the young people. Biotransformation has always been my favorite, because we often do lots of synthetic chemistry by using chemicals, catalysts, solvents, and different kinds. Why don't we use microorganisms? Why don't we use bacteria? Why don't we use fungi? And why don't we just carry out these fascinating chemical changes by using cells? So in our laboratories in Karachi, we have shelves full of bacterial cells, we have fungal cells, we have plant cells and animal cells, and we use these cells as our chemical agent, so-called chemical agent, because it's fascinating. You don't need to use chemicals. The only thing which you need to use is the culture of these cells. And this process is called biotransformation, a very fascinating branch of chemical derivatization, because it provides you very interesting molecular analog. So science is extremely simple. Methodology is very simple. You have, of course, cultures of different kinds, and you have your compound, and you inoculate your cultures with the cells, and then you keep them for incubation for a certain period of time. Once the cells, the bacterial cells or fungal cells incubate the compound, you would be able to observe the changes, but for that you need to extract out your compounds by using simple extraction method, purification. You characterize and you carry out biopsy screening. So primarily the objective of this integrated approach is to use living cells instead of using chemicals. And the advantage of this field is that you get a fascinating new derivatization. Chemical derivatization are very predictable, that only occurs at the active site, but when you use biocatalysts like cells, then when the structures of molecules are very interestingly modified and you get often novel analogs. This is an example of the use of biotransformation, and that we have used for the discovery of aromatase inhibitors. Aromatase is a very important enzyme in the treatment of breast cancer. Breast cancer, as we know, is unfortunately very common disease throughout the world. It is the second major cause of cancer-related death in women. And this is because of the abnormal growth of cells at different regions of the human breast. And most of these breast cancers, 2/3 of them, are because of estrogen. Estrogen is an extremely important growth regulator. It's a hormone which is produced in very large quantities in human ovary, in breast and peripheral tissues. And estrogen is important for the division of cell. So if we want the breast cancer cells not to be divided, then it is very important that we stop the synthesis of estrogen. And this is called estrogen deprivation therapy. And that can only be achieved by inhibiting the synthesis of estrogen. And that is the key of modern therapeutic approaches towards the treatment of breast cancer after surgical removal and after making sure that the recurrence of breast cancer do not really occur again. So treatment option is that you inhibit the enzyme which is responsible of the synthesis of estrogen. This is an interesting slide because here you can see that these are testosterones which are converted into estrogen. Estradiol is in estrogen. This is androstenedione which is converted into estrone. They both are estrogen, and these estrogens are required for the cell division. Without them, cell would not be divided, and you don't want cells to be divided. So therefore, you need to inhibit the synthesis of these two compounds, and that can only be happen by inhibiting the enzyme which is responsible of the synthesis of estrogen. So aromatase is an enzyme which catalyzes the synthesis of estrogen. This is the last step of estrogen synthesis, and in order to inhibit aromatase, you need to develop aromatase inhibitors. And when you have aromatase inhibitors, which are the class of drug, very important class of drug now, then estrogen would not be produced and cell would not be divided. And this is a way of making sure that cancer do not occur or cancer progression is controlled. This is the structure of estrogen. Aromatase enzyme is a complex enzyme, very large molecule. And these are some of the aromatase inhibitors which are available in the marketplace. Our approach was that we wanted to synthesize aromatase inhibitors by biotransformation. And for that, we use various different kinds of steroids because steroids are close in structure of estrogen. And we have then developed a new analog. So the first problem was to where to get the aromatase enzyme. Aromatase enzyme is not easily available. You cannot really order it. It's a very difficult and extremely expensive enzyme to purchase. Some people would produce that for you, but they would charge literally thousands of dollars. And we found a very easy way: human placenta, plenty of it, produce aromatase enzyme, and microsomal cells of placenta contain aromatase enzyme in very large quantities. But we have also, by using recombinant technologies, we also have expressed aromatase in the cell, E. coli. So we have a very pure form of aromatase enzyme and also aromatase enzyme from human placenta. Now, how to measure the activity of aromatase is just to measure the production of acetyl, which is an estrogen, by using ultra pressure liquid chromatography. And I'll just give you one example. I don't want to take much of your time. I just wanted to let you know that this is a synthetic steroid. And here you can see the wonderful chemistry, what happened in biotransformation. So here you can see that this is test electron, and by using two different fungi, Fusarium linea, you can, and the other is amphisolina, you can modify the structure. So you can see that in the structure, there is one oxygen ring, one hydroxy group is introduced, different stereochemistry. And this is the standard drug, exmestine, with which we have compared the structure. So here you can see very well that we were able to produce new libraries of aromatase inhibitors. You can see IC50 value 0.82, which is not different from the standard drug available. But some of the molecules which we have produced were found to be even more active. This is 0.33. And as you can see that this is also as active as the standard drug. The message which I want you to convey here is the following. You can create new chemicals. So we have introduced new analogs of aromatase inhibitors. And these aromatase inhibitors are much more active than what you can see in the pharmacy shelf. And that we were able to do only by using a novel approach. And of course, having a science at the interface of biology and chemistry. So we do lots of interesting synthetic chemistry. We do lots of natural chemistry. We do lots of biotransformation, but as you can see, the major strength of our work is that we have a strong biological background and that we have very experienced scientists working in the field of various fields of biological sciences, from molecular biology to structural biology, from cell physiology to molecular microbiology, whole range of it. So what I wanted to present to you, which is briefly summarized, that it is possible to use natural products and to develop new lead molecule as drugs. And it's also possible to take old molecules and transform their structure by using different combinations of biotransformation. Let me introduce my research group. I have very talented young colleagues, and they are expert in their own fields. By own merit, they know science at a very high level, and they work in combination, in tandem together with me. And we have been very fortunate to have funding from lots of pharmaceutical companies. This is the institute we work. This is famous ITJ Research Institute of Chemistry, which was established back in 1966. Is one of the most famous natural product chemistry institutions in the world. This is a molecular medicine center This is Dr. Taruman Laboratory, who is our patron-in-chief and a very famous scientist in the field of bioorganic chemistry. This is biotechnology, where we do lots of medicinal plant cultivation. This is a nanotechnology center. We have big genome centers. We do lots of genomic work, forensic DNA, and this is a center which we established in recognition of the services of a German scientist. We have our own laboratories at the shoreline of Arabian Sea and also on the mountains. This is our National Institute of Virology, which is a big center doing lots of virus-based research. These are animal houses of different kinds, and of course, lots of. So this is our clinical research unit. We do lots of clinical studies on human patients by using our own hospital. We have a 120-bed hospital. We are right now in the process of conducting phase 1 and phase 3 clinical trial of COVID-19 vaccine. This is our industrial and medical center, our technology incubation centers. And of course, our facilities are among the finest in the world. We have 13 superconducting NMR machines, lots of X-rays and mass spectrometers of different kinds. But I think the most important thing which I wanted to mention here is the quality of people. So there are 17 research buildings, and each building is designed to do different kinds of research. We have our own guest houses and hotel on campus, and we get lots of visitors from all over the world. So we get 200 visitors every year. We have facilitation for female scientists, and of course, lots of other facilities. But I would say this is the actual strength of our institute, the brightest young people, extremely amazing, bright, bold, and beautiful people of Pakistan and from different countries are coming to Pakistan. And we were fortunate to have guidance from Nobel laureates. So we have been welcoming Indonesian students in the past also. We would look forward to have more of them coming to our institute. And our relationship with Indonesia is absolutely phenomenal, brotherly, and we are very close to each other. So thank you very much. Visit Pakistan. It's a beautiful country with lots of culture, lots of diversity, lots of history, and good food, very close to Indonesian food. Thank you.
Okay, thank you very much, Professor Chaudhary, for very, very, very nice presentation. Yeah. Now we have discussion time until 11:55. Okay. If you have some question, please write in Q&A facility. Yeah, before Professor Chaudhary joins, we already have a little discussion, so we will extend in this session. Maybe I would read the one question already appears in the chat room. The question from Mr. Ahmad Fatoni from LIPI. The question will deliver to Professor Gunawan. Thank you for sharing knowledge and lots of information related to quality control of herbal medicine. What are the strategies to develop herbal medicine in Indonesia due to the abundance of our biological resource? Whether herbal medicine research is focused on plants that have been used as traditionally or have never been used previously. And also, Mr. Fatoni delivered a question to Dr. Pradipta. Does the endogenous acrolein only occurs in breast cancer cells, or it can also occurs in other cancer cells, so that it can detect and treat for those cancer cells? Okay, please, Professor Gunawan and Dr. Pradipta answer the question. Maybe next question we'll deliver to Professor Chaudhary, maybe, we hope so. Okay, please.
Thank you for the question. I will try to answer. As I previously talked, the strategy is nice if we in Indonesia do collaboration between university or between research institute, so we can do we have limited facility, not as good as Professor Chaudhary show. We have in our laboratory Indonesia very limited, so that's why we need to collaborate. But platform or ICTI or LIPI should coordinate the research. So the research should be coordinated so the duplication or the same research can be avoided. So what shall we do is depend on the top. I think platform or ICTI should decide what plan should be first. What plan of what traditional plan, what we should do first. So by this, the platform or ICTI can ask certain university to do part of the research, and then we can combine. So by this method, the research will be fast and more efficient. As we know, at present time, many universities do research. Almost university in Indonesia do research on herbal drugs, but I think most of them do the same thing, do the same topic. That is not nice. I think the most important is someone from the Jakarta should coordinate all the research in Indonesia, so we can have a good result in efficient and limited budget. I think that is the strategy that I recommend in my conclusion, and I have I always recommend this in my previous presentation and many seminar. We must do collaboration, but one or platform or ICTI or LIPI should be coordinator. Without that, that's very difficult. Thank you.
Okay, thank you very much for the answers, Professor Gunawan. Unfortunately, Dr. Pradipta already off from this room. Maybe the question will be delivered by email later. Okay, this is one question for Professor Iqbal from Dr. Arthur Lelono from Indonesian Institute of Science. The question is, Assalamualaikum, Prof. Iqbal. Thank you for your presentation. As we know that discovery of natural product took a long time, what is your opinion of using biotransformation? Will it slice the time needed in the discovery of natural products? And can we use deep learning in the structure-activity relationships in the of potent natural product for drugs? Please, Dr. Chaudhary.
Brother Arthur, Assalamu alaikum. This is a very important question. I would say that these are two different approaches. And of course, natural product remain very, very important. But we also know that it is, you're right, that it takes lots of time and lots of efforts to isolate and to get enough quantities of a natural product. But of course, as you know, that lots of new technologies have been developed recently. There are two levels of technologies. Of course, we can do de-replication. We can quickly identify known natural products and new natural products by using hyphenated technique like LCMS, GCMS, LCMS, NMR. And also, recently, there are lots of metabolic engineering is conducted now. It's possible to produce natural products in bacterial cells by identifying genes and then incorporating genes. By using recombinant technologies, one can produce various natural products. There is one approach called self-suspension culture. It's also possible that we have cells of the plant and we grow them in bioreactors, we produce them in large quantities. But you're right that isolating natural products is not trivial, it takes time. Biotransformation is extremely fascinating. It gives you novel analogs and is a very simple science, to be honest. I really would like to encourage people to work in this field because of three reasons. Number one, that you don't need to have very expensive equipment. This is primarily a science in which you need incubator, perhaps some level of, you need to have extraction, you need to have incubations, and you need to have fermenters. You don't need to, even fermentation can be done on simple shaking bath. And second most important reason is that the structure of biotransformed products are not so different from the substrate. So identification of the structure is much easier than isolating a natural product which is completely novel and you don't have no idea about the structure. Often the biotransfer products are analogs of substrate, so their structure is not very difficult to elucidate. The third major reason is you can scale it up any level. You can produce them into any quantities by just scaling it up. You do biotransformation on 1 liter, you can do it 100 liters also, and you can do it on 1,000 liter. It's sustainable science because the microorganisms you can produce as much as you wish. You can recycle the unconsumed substrate back to the loop. So because of these three reasons, biotransformation is still very exciting. But kindly remember that biotransformation would never give you novel, exciting, completely unknown compound. For that, you always have to look towards nature. Even synthetic chemistry would never give you that kind of structural diversity which you see in nature.
Okay, maybe clear, Dr. Arthur? Okay. Yeah, the biotransformation process is very, very interesting. We also conduct in our laboratory, Professor Chaudhary. Yeah. Okay, we move to next question from Ibu Marisa from LIPI to Professor Chaudhary. The question is, it's a nice presentation. Could you share about the results of your institution research related with the implementation? It have been done communication with some pharmaceutical industry, because sometimes in Indonesia, we have difficulties when we want to commercialize our products. Maybe you can suggest or something.
Yeah, because Marisa is also a very important problem which we have pointed out, an extremely important bottleneck which we all face. Now, there are two approaches. One is that if you wish to develop a pure natural product as an ethical pharmaceutical, then you have to go through all clinical trials and everything. And that requires lots of money and lots of efforts, lots of time. We have been doing it in case of one anti-epileptic drug, which is called ISAC. This is in phase 2 clinical trial. This is the 11th year of development. It took about 11 years for us to reach to, and for that we got funding from a Canadian company, and that is a pure natural product, a natural product which we have then synthesized because you cannot get them enough quantities from the plant. So this is one approach. The second approach is, which I strongly believe is the one which we should really do, is for curiosity, you can always go and isolate compounds, study the mechanism of action, but always remember that if you would like to develop a pure natural product, then the cost is extremely high, because purification of a natural product in required pharmaceutical grade is extremely expensive. So I would always recommend that we work on composite medicine. We work on fractions, standard fractions. we prove their efficacy, and we prove their safety. And that is a drug by all definitions. So for us in developing world, the best way of moving forward is to develop often very symptomatic, and there is no medicine available which can provide them a complete relief from some symptomatic issues. So this dietary supplement, which is now internationally patented, and I would be happy to deliver a lecture about that separately, offline, is developed in collaboration with Searle. But again, this is a combination. This is not a pure natural product. This is a combination of drug and a combination of dietary substances, plants, and by using different.
Okay, thank you very much. Okay, we move to next question from Dr. Abdi Wira. The question is, thank you for a nice lecture, Professor Iqbal. I'm interested with your research about antimicrobial because it is somewhat similar with my research. I want to ask you about the best way to develop such antimicrobial compounds to be oral drug instead of topical application. In your opinion, what's the best model in animal that can be used for this purpose? Okay, please.
Okay, Professor Andre, this bacteria, this so-called infectious model are the easiest to develop. That in among all the models, the best models which we can develop for topical application, of course, you need to have, if you are developing a topical anti-infectious agent, you can always develop topical infection. So you need to immune compromise because often these rodents do not really, they are so hard that it's very difficult to develop a topical infection on them, and you need to immune compromise them. So you need to give them some amino suppressant, and then only you would be able to develop candida infection or staphylococcus aureus infection or pseudomonas infection. So topical application, topical infectious models are very, very easy, but you need to suppress the immunity of the animals also. For systemic model, which is required for oral or injectable antimicrobial, then it's called bacterial load, so-called microbial load model, in which you inject bacterial cell in certain numbers, and then you measure the bacterial load by using flow cytometry. This is a time course study because with time you would be able to see that the bacterial cells multiply, you have more of it, and often you may not really see any infection. I mean, you may see lots of bacteria in the work always start from in vitro screening, 96-well screening, in which you have standard cultures, because that is the beginning of the work, and only then when you see the activity demonstrated in vitro, then you go to the animal model.
Okay, Pak Abdi. Okay, thank you very much for explanation, Professor Chaudhary. Your presentation is quite interesting. Still more two questions in the chatting room. Okay, next question come from Ibu Sofa Fajria from Indonesian Institute of Science. The question is, thank you very much for nice presentation, Prof. Chaudhary, regarding integrating natural and scientific approach. Usually in natural product, we only got a little amount of compounds, especially for new compounds. Could you give us suggestion how to get efficient of isolation or scientific of natural products? Thank you very much.
Okay, I agree with you. It is a problem, and that is a problem which would always be there, because natural products are often, secondary metabolites are often available in very little quantities. So I honestly, I do not have a solution, because if you harvest plants from the wild, and then you try to isolate compound from that, you literally destroy forests. So I would not recommend that you go to collect a plant from the wild and then try to isolate large quantities of natural product. The best is ex situ cultivation. If you can carry out ex situ cultivation, a plant can be grown it can be cultivated out of wild. That is one source, and that the Chinese are trying to do that also, because the Chinese are growing many of these medicinal plant ex situ. So this remain a problem. Second is, of course, you can produce them in enough quantities if you develop alternative methods. One, as I mentioned, metabolic engineering. It's possible to identify the gene of enzymes which are responsible of catalyzing the production of that, and then modify those genetic engineering by using genetic engineering and produce them in large quantities by E. coli, Saccharomyces. Many of the natural products have been produced. The third way is biomass. So you produce these natural products in cell culture. Instead of getting them from the wild, you can grow them in bioreactors. But having said that, this is remain a problem. I would advise you to read a very interesting paper on the production of Taxol. The similar problem was faced when Taxol was identified as an anti-cancer drug, and they wanted to have only 10 grams of that, and they could not produce because Taxol is in the bark of Taxus braggifolia and need to beat the bark of a tree to produce them enough quantity. So they have then developed alternative method. But eventually, at the end of the day, if a natural product is...
Thank you very much, Prof. Chaudhary. Nice to meet you in here again.
Thank you.
Yeah. Okay, thank you, Prof. Andrea.
Okay, thank you very much. You're welcome. Okay, we move to next question from Dr. Andri Frediansyah from Indonesian Institute of Science. The question is, thank you for our awesome presentation of Professor Chaudhary. What do you think about the discovery of natural bacterial product at this time or recently? Why most people rarely find the new or novel structure or even new natural product classes, even though genome mining is starting increasing? This evidence could be seen in the Journal of Antibiotic published by Nature. The most researchers only found derivatives or analog. During the antibiotic era, we knew that many antibiotics had been discovered from bacteria. How about in Pakistan, related microbial natural products? Please.
Okay, again, a very interesting question. You see, most of the anti-infectious agent, antimicrobial agents which we use today are of natural origin. So it's very obvious that majority of the natural product, majority of anti-infectious agents which we see used today, either from bacterial sources or from fungal sources or from plant sources. And so they remain a very important source of it. Problem with the antibiotic is that not many people are working in the field also now. Pharmaceutical companies do not work in the field of anti-infections, because of a very simple reason. They spend $2 billion, $2.5 billion. They know that within few years the resistance is developed, and often before they get return of the investment, their antibiotics is already useless. So not many pharmaceutical companies are working in the field of antimicrobial drug discovery. In Pakistan, we in our institute we are working on the discovery of antimicrobial agents, but not antimicrobial agents sole. We have now a separate laboratory for resistance reversal agents. So what we are trying to do is the following, that we're trying to identify compounds which help existing antibiotics to function. We consider this approach is a very interesting approach because development of antibiotic from the scratch is expensive. Augmentin is a one classical example in which antibiotics is supported by a beta-lactamase inhibitor. So it's possible to develop antimicrobial formulation by using existing antibiotics and resistance reversal agents. And that resistance reversal agents are often you can obtain from the natural sources. Now, again, how much you can obtain is a problem itself. But my response to your good question is that this is an extremely important area. This is an area on which human survival based. Resistance of microorganisms is a great challenge for all of us, and we all must.
But if possible, I will have one simple question, Professor.
Please.
Okay. This is only a simple question. During your long career as scientist, which research very special for you? Or maybe your research result special or something, and why you think that is special? Okay, please.
You see, I've been working in this field since last 100 years. You know, I'm just so old now. But and it really excites me even today also. When I isolate a new compound, when my student brings something new for me, I feel the same excitement as I used to feel centuries ago. But one research which I feel I have greatest satisfaction is the one which I have done with the skin diseases. You know, when you will come to Pakistan, I'll show you that certain regions of the country, there are a tropical disease called leishmaniasis. It's also in Indonesia. It's very, very common disease, but in Pakistan, it's unfortunately very endemic, and many regions you find this sandfly producing, transferring this vector, and people have lots of patients, and often they live with it for the rest of their life. It's not fatal. It doesn't kill a person, but it really have a problem, especially for young girls, because I've seen young, beautiful girls with scars on their face, their noses, and I really felt bad about it. Now, what we have done was absolutely a very simple science, nothing exciting. What we have done was that we asked them what they use, and then, you know, out of this ethnobotanic survey, we identified that in some region of the country, people have been using simple natural product for the treatment, and sometimes they were able to treat that also. And on the basis of those ethnobotanic formulation, we developed a natural product-based ointment, which is now available in the country for leishmaniasis. And over 2,000 patients, clinical trials were conducted on 2,000 patients, because this is a very common disease. So a very simple science gave me lots of satisfaction. Of course, I have done lots of complex sciences. I have many studies conducted at a molecular level. But at the end of the day, what you can see happening to the people, and if you can help them in some way, ultimately that gives you the greatest satisfaction.
Okay, thank you very much, Professor Chaudhary. Yeah, I met 13 years ago with Professor Chaudhary in Bangkok, and Professor Chaudhary asked me to move to Pakistan, but I don't know what the reason not reliable for me at the time. Okay, thank you very much, Professor Chaudhary. And also time is over. I would like to say thank you very much for all keynote speaker, Professor Gunawan, Dr. Pradipta, and also Professor Chaudhary. Thank you very much. Okay,
thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Okay, I return back time to the organizing committee. Ibu Marisa, please.
Thank you, Professor Dr. Andrea Agusta, for the nice presentation today. Dear our speakers and our moderators, and also all of the participants, thank you for the time with a nice presentation today and also fruitful discussions. We will have a break time from 12 until 1 p.m. But before break time, I want to tell about our next schedule. Next and at noon, we have 2 parallel sessions. The first parallel session will be start from 1 until 3 p.m. The first parallel session will be divided into 3 rooms. Room A about natural product and traditional medicine will be moderated by Dr. Sofa Pajriah. The second room will be discussion about pharmaceutical technology, drug delivery system, analytical chemistry, and chemical engineering with the moderator Siti Nurul Aisyah Jenny, PhD. And the third room will be discussed about food technology and functional food with the moderator Hakiki Melani, PhD. Ladies and gentlemen, the second parallel session will be start from 3:15 p.m. until 5 p.m. and also will be divided into 3 rooms. The first room about natural product and traditional medicines and will be moderated by Abdi Wilaseptama, PhD. The second room will be discussed about analytical chemistry, chemical engineering, and will be moderated by Dr. Indri Badriah Adilina, Master of Engineering. And the third room will be discussed about food technology and functional food and will be moderated by Khairunisa, PhD. We hope all the participants will be joined in each room until end of this day. And the participant will automatically enter each room according to the schedule that you can read in the program book. Okay, we hope all of the participants enjoy the symposium. See you tomorrow. Bye-bye. Assalamualaikum warahmatullahi wabarakatuh.
Kita boleh live ya?
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. It's called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food. Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of good manufacturing process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food. Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food. Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are standardization of food processing technology. The packaging technology has received a certification of good manufacturing process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research. Scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens. Bogor Botanical Garden, Barium Museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers, for UNESCO, and has 3 international centers, namely Asia Pacific Centers for Eco-Hydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future.
Such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how in the institution? The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The packaging technology has received a certification of good manufacturing process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817 with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces, 100 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research. Scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens. Boko is the 3rd largest herbarium museum in the world. And Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers. 2. Accrediting scientific journals. 3. Studying and evaluating biodiversity. Establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO and has 3 international centers, namely Asia Pacific Centers for Ecohydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for all the participants for joining back after your break time, and welcome back to the parallel session of the 6th International Symposium of Applied Chemistry. So this parallel session will cover the topic related to the food technology and functional food. So first, let me introduce myself. I'm Hakiki Melani from Research Center for Chemistry LIPI. I will be the moderator for this parallel session that will be consist of 8 presenters. So, but before we start the presentation, I would like to explain several points regarding this session. So we will have the two part of this session. The first part will consist of 4 presenter, and the presentation will be delivered by the pre-recorded video with given time of 10 minutes. And after all of the 4 presenters give their presentation, we will have the discussion session for about 20 minutes. So during the discussion, you can raise your hand to ask the question, or you can chat through the chat room. So without further ado, let's begin this parallel session with the first presenter. I think all of the presenters already here. So the first presenter will be delivered by Mr. Kurniadi from the Faculty of Animal Technology from Gajah Mada University. Okay, so to the co-host, could you please share the video? Thank you. To the co-host, sorry, we cannot hear the video. I think we have some trouble, technical trouble. So we will fix it as soon as possible.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. I will present our research about fermentation of amylolytic cheese and lactic acid bacteria to improve the quality of modified cassava. Cassava, money hot as cleanse. Fresh cassava is easily damaged, contain high water and anthocyanin, especially hydrogen cyanide. Fermentation is important technique to make cassava safe for consumption. From cassava fermentation, especially in Yogyakarta, such as growol, tape, tiwul, and gatot. Natural in fermented cassava, there is lactic acid, bacteria, and this the info in biochemical composition chain. Modified cassava fermentation now is one of the fermented cassava products that is being developed in Indonesia. The use of inoculum for mock-up fermentation is important to ensure the quality product according to the standard. This study aims to select microbes with amylolytic activity as an inoculum in the fermentation process of cassava and potentially reduce the content of cyanide acid in the final product. Materials: the traditional fermented cassava used for simple ingest isolation. Tape, tiwul, gatot, and ragi tape obtained from Demangan Market and Wonosari Market, Yogyakarta, Indonesia. 21 of LEV isolate were microbiological laboratory collection in PTWA LEV. LEV elicit from fermented cassava such as gatot, tiwul, gaflek, and modified cassava present products. Local variety of cassava root obtained from the Wonosari Market, Gunung Kidul, Yogyakarta, Indonesia. The method: cheese isolation using chloramphenicol cheese glucose plus media and incubated for 72 hours at 24 derajat Celsius. Qualitative amylolytic assay, quantitative amylolytic assay, cassava fermentation, molecular identification, biochemical identification, morphology analysis using scanning electron microscope, statistic analysis. The result: quantitative amylolytic activity, lactic acid bacteria G6 elicit from cathode and GSTR7 excluded from ragi tape has the highest amylolytic activity. The parameter of cassava fermentation using GSTR7 and lactic acid bacteria G6 inoculum. Cassava fermentation process with inoculum result: higher on ILV population, lower on coliform population, lower on VI value, higher on temperature, lower in cyanide content, higher in cyanide reduction than weighted inoculum. Cyanide content in fermented cassava process, fermentation type E and fermentation type B. Type E has and reduction the 52.19% and B 32.38%. Type A with inoculum and type B without inoculum. Biochemical and molecular identification. The result of identification using the IPA 50 CRL show that the G6 isolate the head 99.6% of similar with Pedococcus pentosecius. The 16S RNA application result of ILEV G6 isolate obtained sample. PCR products visualized using a cell documentation system with 1,500 P4 was identified as Pedococcus pentosecius strain BSS 1375, 100%. The result using a VI 20 CIUX gets shown that the TR7, this slot had 95.8% similarity with Candida tropicalis. The VCR result from the 20 ISRRNA sequence after assembly and PLHD nucleotide show that TR7, this is slot with Candida tropicalis strains, and ITCSK1 with 99% of identity. The morphological picture of the species, lactic 8 bacteria Pediococcus pentosus G6 slot is a gram-positive bacterium that has cell size of 0.5 until 1.2 times 1.0 until 10 micrometer Shock scale and everything in fires. Which Candida tropicalis, the ISA7 has official and branching like the genus Candida in general. Pediococcus pentosus is LV which was commonly found in final foods products such as rice fermentation, tempe cassava fermentation, corn, mild and sorghum fermentation. Candida tropicalis with aminolystic activity in cassava fermenting water, fermented cassava meal. Conclusion: GHTR 7 and LVG6 isolate show the highest amyloidic activity 0.06 to 4 units per milliliter, 0.06 to 7 milliliter. Cassava fermentation product using GHTR7 and BIL-G6 result 23.06% and Hargensen reduction higher than without inoculum, 32.32%. Lactic acid Candida tropicalis CR7 potential use for inoculum in cassava fermentation, process into improve cassava quality as function food. Thank you very much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Mr. Kurniawan, for your nice presentation. Okay, we will continue to the second presenter, Ms. Susti Istiqomah, from Research Unit for Natural Product Technology LIPI, with the title Biomodified Soybean Meal as a New Protein Source for Food. So, co-host, could you please share the next video?
Good afternoon, ladies and gentlemen. First of all, I would like to thank the ISAC committee. My name is Susti Istiqomah, and today I will present our study result with the title Biomodified Soybean Meal as a New Protein Source for Food. The research team consists of Research Division for Natural Products and Knowledge Meal Processing. By the fermentation process, we obtain the fermented soybean meal, and then we count the microbial population and plot the growth curve. And we determine the anti-nutritional factors such as phytic acid and aflatoxin, and then we determine the nutrients composition. And the last one, we analyze the data. This is how we prepare the microbial culture using MRS agar and MRS broth medium. Through this process, we will obtain the inoculum of Lactobacillus plantarum A1E. And then we add this inoculum into sterile soybean meal, which previously added with distilled water. And the fermentation process performed during 72 hours. And through this process, we will obtain the fermented soybean meal. During the fermentation, we also count the microbial population every 24 hours. And then we count the colony based on the total plate count method. In order to determine the phytic acid content, we use the qualified assay kit from Magazyme, and then we read the absorbance of the final solution using spectrophotometer at 655 nanometer. Meanwhile, to determine the aflatoxin content, we use the Aflaquan aflatoxin kit from Robert Lab. And to read the absorbance of the solution, we use the ELISA reader at 450 nanometer and 630 nanometer. And then we also calculate the nutrient composition of the unfermented and fermented soybean meal, consists of moisture content, ash, crude protein, crude fiber, and lipid, based on the AOAC method. We also analyze the data using analysis of variance followed by Duncan in order to determine the significance of microbial population between treatments. We also use the t-test to determine the significance of phytic acid, aflatoxin content, and nutrient composition between treatments. These are the results of our study. As we know that Lactobacillus plantarum is considered as grass, generally regarded as safe. So it is currently used as a probiotic, as silage inoculant, and also for food fermentation. Based on table 1, we could see that the population of Lactobacillus plantarum A1E increased during the fermentation until 48 hours. And the population stable between 48 and 72 hours. And then we convert this data into log 10 CFU per milliliter, and then we plot on this growth curve by subtracting the data population from 24, 48, and 72 hours to the 0 time. From this graph, we could see that this is the lag or adaptation phase. This is the log or exponential phase until 48 hours, and this is stationary phase from 48 until 72 hours. We also determine becomes myoinositol as main product, release the inorganic phosphor, and release a metal cations such as calcium, iron, and zinc. Therefore, the phytic acid content decreased. Meanwhile, the aflatoxin content between treatments did not significantly different, but there was a tendency a reduction around 6.12% of aflatoxin content. Another research reported that the Lactobacillus plantarum strain LB7 and LB9 isolated from sorghum perform a poorly aflatoxin B1 binding abilities only 2.1 and 5.2%. While our previous study reported that Lactobacillus plantarum G1 addition in the aflatoxin contaminated diet decreased the aflatoxin content. This is the proposed to activate the GSTA3 through the NRF2 signaling pathway. So it stimulates the glutathione conjugating activities and induce the detoxification. From this graph, we could see the comparison between nutrient composition of unfermented soybean meal marked with the dark blue and light and fermented soybean meal marked with the light blue. As you can see here that the ash content, fiber content, and protein content increased significantly. Meanwhile, lipid content was stable. Another researcher reported that protein content in the fermented soybean meal by Bacillus subtilis was increased due to the degradation of allergens into peptides. Thereby, peptide can be easily absorbed and transported within an organism. As I previously mentioned, the phytic acid form a complex with the protein and starch. By addition of microbial producing phytase, then it will break this formation complex. So the protein, carbohydrates more available and more degradable, thereby increase the nutritional value of SPM. From this data, we conclude that the biomodified soybean meal represents an improvement in nutritional quality and becomes potential raw material as a new protein source for food. Thank you very much for your kind attention and good afternoon.
Good afternoon, ladies and gentlemen.
Thank you for Miss Lusty for the nice presentation. And we will move to the third presenter. With the third presenter will be Dewi Desni Lasari from Research Center for Appropriate Technology, LIPI, with title Using Kefir Glucomannan to Modify the Diversity and Composition of Sesum Bacterial in Sprout Dowley Rats with Metabolic Syndrome. The co-host, you can please proceed with the next video. Thank you.
Everybody, my name is Dewi Desni Lasari. I'm from Research Center for Appropriate Technology, and in this occasion, I will present my paper with the title is Using Kefir Glucomannan to Modify the Diversity and Composition of Secum Bacterial in Sprague-Dawley rat with Metabolic Syndrome. As we know that the lifestyle, diet are the important factor that leading to the rapid occurrence of metabolic syndrome, with the characteristics such as heart disease, lipid problems, hypertension, and type 2 diabetes. All of these will change the composition of gut microbiome that influence the health of the body. But many researchers assume that administration of probiotic and prebiotic will increase the microflora, which is beneficial to the health of the body. In this research, we use kefir as a probiotic and glucomannan as prebiotic. We hope that will promote the growth of good microflora. And the aim of this research was to know the effect of supplementation kefir and glucomannan to the diversity and composition bacteria in the rat Sprague-Dawley with the metabolic syndrome. For material and methods, first we produce the kefir and then do the animal experimental and then collect data such as the body weight, feed intake, and secum bacterial analysis. This is for the result, the number of species, diversity index, and frequency distribution. From this data show that in statistical analysis, so wasn't significantly different among the treatments. And this is for the data, the composition bacteria in secum. This is so that in the blue one is lactobacillus, red is bifidobacterium, and green is for lactobacillus. This is lactococcus, and purple for the bacteroides, and this is for clostridium. From this picture, show that the mostly found the bacteria was the lactobacillus, bifidobacterium, lactococcus, and bacteroides, and also clostridium. In this figures also show that using kefir and kefir glucomannan will increase the number of lactobacillus, and it's also with the bifidobacterium. If we saw, then the rat with metabolic syndrome not administered with kefir and kefir KM. And in this figure also show that the clostridium and the bacteroides with the using kefir and kefir GM also will decrease. And this is our finding that indicated the supplementation of kefir and kefir glucomannan will support the beneficial microflora in the gut. And this is the data for the body weight of the rat. This is so that there is no significantly difference among the treatment. It means that administration of kefir and kefir KM would stabilize the body weight of the rat. And this is the data for the average feed consumed per day in group treatment. This is also so that wasn't significantly different between the treatment. It means that uh, indicated that supplementation kefir and kefir glucomannan could stabilize the body weight gain with inhibition the rate to increase the feed consumed. And all of this with the conclusion that kefir and kefir glucomannan didn't affect diversity, but it is able to modify composition bacteria and digesta by increasing lactobacillus and bifidobacterium and suppressing clostridium and bacteroides. And then kefir and kefir glucomannan can keep the weight body and feed intake in rat with metabolic symptoms. I think that's all. Thank you. Good noon, everybody.
Thank you for Miss Dewi for the nice presentation. And we will move to the fourth presenter for this first part session. The next presentation will be delivered by Mr. Andi Febricianto from Research Division of for Natural Product Technology LIPI. The title of his presentation is Local Buffalo Milk Kefir Characteristics Affected by Fermentation Process. To the co-host, please proceed to the fourth presenter video. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. Thank you very much for attending this symposium today. And thank you for the moderator of the sessions. So today I am Andi Febri Santosa from Indonesian Institute of Sciences located in Gunung Kidul, Yogyakarta. Would like to share one of our research entitled Buffalo Milk Kefir Characteristics Affected by Fermentation Process. As background of this study, we know that functional food is known or defined as the food that not only supply the nutrition for the human, but also have a beneficial effect due to the particular component that may promote the health of the human. Kefir is known as one of the milk product, processed milk product that based on the previous study, kefir so may affect to suppress the pathogen, accelerate digestive system performance, reduce cholesterol level, lowering blood pressure, improve the human immune system, so that kefir may promote as one of the functional food. One of the resort of the kefir is the milk, and one type of animal or livestock that produce milk is buffalo. In Indonesia, the milk from buffalo or buffalo milk is widely used for the traditional food product, especially in Sumatra Island, Indonesia. But there are still lack information or utilization of buffalo milk utilization for the kefir production and its beneficial effect. So the objective of the study is to evaluate the effect of the local buffalo milk fermentation using kefir grain on its chemical, microbiological, as well as organoleptic characteristics. Actually, this study is just the first step of the whole study, which for the whole study, the next step is the beneficial effect study of the kefir produced from the buffalo milk. We have two general steps. The first step is buffalo milk kefir productions. The milk was pasteurized and continue with cooling, and then inoculate with the 0.5% kefir grain. After that, we incubate the milk that was included with the kefir grain with different incubation time as a treatment, which was 12 hours, 24 hours, 36 hours, and 48 hours with the running temperature. And for the microbial characteristic, we evaluate the total active bacteria, total yeast, and for the sensory evaluation, we evaluate the texture, flavor, and acid characteristic. Yeah, as a result from the experiment, it showed that the first figure 1 and figure 2 in this slide was the total acid concentration and the lactose concentration. We found that this was significant effect of the fermentation time to the total acid and lactose. The lactose was reduced and the total acid increased. So it was likely be to the lactic acid bacteria activity to produce lactic acid metabolic product. So the highest total acid reached by the treatment with 48 hours. And for the lactose itself, the lowest lactose reached by the 48-hour fermentation time. This condition is followed by the increase of thickness texture formation to the product as a result of the fermentation. So inoculation of kefir grain shows the fermentation was going on in product. Continue to the fat and protein content in the product. So we found that to a significant effect of the fermentation time to the fat and protein content. As we can see here, the lowest fat contained by the treatment of 36 and 48 hours fermentation time. And for the protein itself, the highest protein contained by the 36 and 48 fermentation, 48-hour fermentation time. So it was likely due to the microbial biomass contribute to the protein availability in the product. So the protein will be increased in the final product. As a result of microbial activity, actually microbes will produce lipase enzyme, and then the enzyme will hydrolyze the fat on the product during the fermentation process, so that the fat content will be lower. Will be lower in the treatment with the longer fermentation time. The next one is alcohol and carbon dioxide concentration. So for the alcohol itself, for the alcohol concentration, after 48, the treatment with 48-hour fermentation showed the highest level of alcohol, and as well as carbon dioxide. The carbon dioxide concentration in the product reached the highest level and with the product with 48-hour fermentation time. So the significant effect on alcohol and CO2 concentration occurred in this study. Actually, this production of alcohol and CO2 concentration result by the yeast activity. So this condition also contribute to the product sensory quality showed by the soda sensation if we test the product. The next result is the microbial characteristic. We see here lactic acid bacteria and total yeast. Lactic acid bacteria and yeast availability increase along with the fermentation time, and lactic acid bacteria actually significantly increase after 24 hours fermentation, and yeast significantly increase after 36-hour fermentation time. In here, we can see that symbiotic relationship between lactic acid bacteria and yeast during the fermentation process. The lactic acid bacteria will utilize the lactose to break it down into the simple sugar, like lactic acid, and the simple sugar will be utilized by yeast to grow. And the growing of the yeast will result the biomass or biofilm. I'm sorry, will result the biofilm formation. We may filtrate the kefir grain, separated kefir grain with the milk. So with the kefir product, I'm sorry, and then the kefir grain can be utilized again for the inoculum. And next one is the organic and sensory characteristic of the buffalo milk kefir with the different incubation time. Here we can see that the product with fermentation time 12 hours and 48 hours were less preferred by the panelists, shown by the less value of the preference. And we test the texture, flavor, acid, and overall characteristic. However, the kefir with 24 hours fermentation time was considered the most preferred by the panelists. So the less preferred panelists here could be due to the acid test in kefir. We can see, as we know, that Indonesian people have mostly less preference to the strong acid in the food product. So that's why we'll show the less value. The flavor produced in the kefir actually related to the fatty acid composition result from the fat decompositions. As we can see, the previous result that the composition of fat reduced in the after the 24 hours fermentation time, and the lipolysis occurred in the fermentation process of the fat component in milk produce free fatty acid, which are precursor of flavor compounds such as alcohol, methyl ketone, and ester, and it contributes to the flavor formation. So the conclusion of this early study is the fermentation time affected to the buffalo milk kefir characteristic in terms of increase the total acid, protein level, alcohol, as well as carbon dioxide concentration. Fermentation process might reduce the fat level and lactose content of the final product. And after 24 hours of incubation time can be considered for producing the buffalo milk kefir with preferable for the consumer. This is the end of my presentation, and I'll give it back to the moderator. Thank you very much.
Okay, thank you for all the presenters that have been delivered the nice presentation. So we will now come to the discussion session. During the discussion session, we are expected that all of the presenters that have been present will prepare their PPT if there's like a further question that need to be explained. So the first question comes from Miss Lusti Istiqomah and directed to Mrs. Dewi Desnilasari, the third presenter. The question is, how do we know that the enhancement of lactobacillus due to kefir induction on the growth of lactobacillus, which is naturally present in GIT and not from the addition of kefir, which contain lactobacillus bulgaricus? To Mrs. Dewi Desnilasari, you may unmute the speaker and answer the question. Thank you.
Okay, thank you. This from Miss Lusti. Maybe you could explain with the GIT with the. Huh? Oh.
GIT mean gastrointestinal tract.
Oh,
Lactobacillus is one of micro.
okay.
Okay.
This is, may I share the screen?
Yes, please, you can share the screen. The co-host, please help the presenter.
I can.
You
I
cannot
I still cannot.
cannot share a screen share.
No problem. Maybe yeah. I will tell more. Yes. In my actually, this is the research is only a prediction. Yeah. So that the prediction for the what is the actually the mostly bacteria in the gut microbe, the second of the, what is it? Yeah, because this I'm using the TRLP. It's just only a prediction of the bacteria that in the second. But if you see my slide in the composition bacteria, this is I just compare with the rat that has metabolic syndrome that doesn't administration with kefir and kefir glucomannan. So this is in this actually from the prediction show that that lactobacillus is also lactobacillus increase with the administration of kefir and also with kefir glucomannan. And then I think that I don't know that this I cannot, what is it? I cannot sure that it's I cannot make sure that it's because the administration of the kefir, because I just predict what is will what the composition bacteria will be if we administration the, because if in kefir there are lot of lactobacillus that or I don't know that if probiotic sometimes we administration is already known, yeah, already known that maybe just only one species we use, but in kefir we use a lot of most or is it complex bacteria that is also there are all lactobacillus and pediococcus also, and then we cannot assume that and we cannot assure that there is my lactobacillus in kefir is change the, what is it, the composition bacteria in secum, because what is it, we just spread what is it, describe and catch um, the data only to predict what will be the bacterial composition and that secum. I think that's all.
Okay,
Yes.
thank you, Mrs. Dewi. To Miss Lusti, do you have any further question, or are you already satisfied with the answer?
No, thanks. Thank you.
Okay, thank you. The next question is coming from Miss Emma Damayanti, also to the Miss Dewi. The question is, please explain in more detail about change in the microbial population in mice which was treated by glucomannan. Which microbial groups were dominant, change, or increase? to Miss Dewi Desnilasari. You may answer this question.
Okay, thank you. As I explained before, that there are increase in lactobacillus and also in bifidobacterium. Maybe if we see the picture, we will see clearly because I can share the screen so that I cannot explain more. Just only say that I already said that the lactobacillus and bifidobacterium also increased, but in the other hand is the Clostridium and Bacteroides is has a decrease. And what point? What is it? What the number is a percentage of the increase and decrease? I'm not. What is it? I'm not tell in the presentation. Actually, the lactobacillus is increased for 14.61%, and for bifidobacterium is 3.7%, and for the decrease of Clostridium is 38.15%, and for Bacteroides is 22.51%. I think that's all.
Sorry, Miss Dewi, maybe you can try again to share the screen. I think it's already allowed by the co-host, the settings. Maybe you can try again to share the PPT screen.
This is already seen or?
Yeah, already seen.
Yes. And this is this is already see that this is the rat, the combustion bacteria in rat that with the metabolic syndrome, but not administration with the kefir and kefir glucomannan. And this is the picture for the rat that with metabolic syndrome. Oh, yes. I'm sorry, I'm not slow. Yes, yes. This is maybe too. The number is too little, yeah. This is in this this is blue one is the for lactobacillus, and the red one is for bifidobacterium, and the green one is for lactococcus, and the purple is for Bacteroides, and this is for the Clostridium. This is the same pattern. This is also same pattern. And then from this data is so that compare with the red with data with the red, there's an administration with kefir and kefir glucomannan, so that the lactobacillus more bigger and more than with the red that not administration with the kefir and also the kefir glucomannan. But we cannot sure that the lactobacillus in here and also in here is from the kefir that we administration, because there is no we just using the TRLP, so it's just only what is it, the based on the what is that fragment of the bacteria.
We still have 5 more minutes. If you have another question, you can chat, or maybe you can also ask directly in this forum by raise your hand. Is there any question? If I would like to ask a question to the first presenter, Mr. Kurniawan. I would like to ask a question to Mr. Kurniawan regarding the use of the amylolytic yeast and lactic acid bacteria that can improve the cassava quality as a functional food. Could you explain more in which aspect that the cassava quality can be improved by using this inoculum? Is it in terms of physicochemical properties or maybe the chemical, a bioactive chemical? Could you please explain to Mr. Kurniawan? Thank you. To Mr. Kurniawan, you can unmute first your speaker.
Okay, thank you. Okay, maybe my thing, Bu Emma can answer about this action. Please, Bu Emma. Hello, Bu Emma.
Hello.
Hello.
Yes.
Yeah, I already open.
Hello, Bu Emma.
Hello, hello, Miss Emma.
I can share our presentation.
Yes, you can share the screen.
Mhm. Okay, thank you for the question. In this research, oh sorry. In this research, in this part, we focus in the microbial population, lactic acid bacteria and coliform as a contaminant. Oh, still okay. This one. We only focus in total lactic acid bacteria and total coliform because this is the beneficial bacteria, and this one coliform is a pathogenic bacteria in as a contaminant in mocaf production. On biochemical, we we just study on cyanida content because this is the main toxin in cassava, and we focus on cyanide content, and we can show that there is a reduction cyanide content after fermentation using inoculum. But in other part of this research, we evaluate biochemical properties like carbohydrate, protein, and other parameter, and improve, especially in protein content and protein content and carbohydrate content, such amylolytic, amylosa and amylopectin. And then we found that there is improved characteristic in what is derajat putih white derajat keputihan derajat warna
maybe
ya Yeah,
whitening degree.
Yeah, maybe whitening degree.
Whitening.
But not in this research. I'm sorry. Okay, it's clear maybe.
Oh, okay. Thank you. Thank you for your explanation.
Thank you.
Okay, so we will finish the first part and we will continue to the second part of the presentation. So the next presenter will be Miss Eni Soliha from Research Center for Appropriate Technology, LIPI, with the title The Influence of Coffee Cherry Fermentation on the Properties of Cascara Arabica from Subang, West Java. To the co-host, please share the video of Miss Eni Soliha. Thank you.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. My name is Eni Soliha. I'm from Research Center for Appropriate Technology, LIPI. Now I will talk about the influence of coffee cherry fermentation on the properties of Cascara Arabica, Subang, West Java. Cascara was prepared from coffee cherry husk. Coffee cherry husk is byproduct of coffee production. Many researchers have reported that coffee cherry has rich of polyphenol compound such as chlorogenic acid, protocatetric acid, gallic acid, and also has high antioxidant activity. Nowadays, fermentation technique is applied for coffee production process to improve the flavor, the taste, and the quality requirement from the customer. This fermentation process will influence the characteristic of coffee cherry husk as raw material of Cascara. In the study, we investigated the fermentation of the characteristic of Cascara, include color, phenolic content, flavonoid content, caffeine, antioxidant activity, and sensory acceptability. This is the experimental method. We obtain the Arabica coffee cherry from Subang, West Java. And the next step is sortation to remove the overripe coffee cherry and to remove the impurities. And the next step is fermentation. We did fermentation in 3 conditions. The first is spontaneous fermentation using fresh water. And the second, we use Lactobacillus plantarum. And the third, we use Saccharomyces cerevisiae. The fermentation have been done in a room temperature for 24 hours. And next, we peel the coffee cherry, and then we dry, and we mill the dried husk, and then we package in the tea bag, and we obtain or we result the cascara. The cascara extract or the cascara infused water, we characterize, include color, total phenolic content, spectrometer, flavonoid content, caffeine, antioxidant activity, and sensory analysis. This is the result of our research. The first is the color of cascara extract or cascara infused water are yellowish and redness. And the yellowish, it's influenced or affected by the chemical compound like epicatechin, and the redness color resulted or formed by rutin and beta-carotene and the other organic compound. And from the diagram, we can see that all fermentation methods did not influence significantly color of cascara infused water. And for the total phenolic, we can see that the fermentation using Saccharomyces cerevisiae has the lowest value of total phenolic compound. Based on the report from Pushwiati et al. 2013, Saccharomyces cerevisiae produce extracellular enzyme, including amylase, which can hydrolyze the bonding of amylopectin and change the sugar become alcohol. And based on the Muller 2013, phenolic compound conjugate with amino acid and glycoside. So it was possible the hydrolysis reaction can increase the solubility of glycoside and phenolic compound in the water. This is for the flavonoid content. All fermentation methods did not affect the flavonoid content of cascara infused water. But we can see that the value of fermentation using Lactobacillus plantarum and Saccharomyces cerevisiae lower than spontaneous fermentation. For the caffeine content, we can see that the lowest value resulted from spontaneous fermentation, and the second is for Saccharomyces cerevisiae. Hartin-Ningsi 2018 reported that in the spontaneous fermentation may consist many kinds of microbes, but the dominant were lactic acid bacteria and yeast, which could produce pectinolytic and proteolytic enzyme. Furthermore, the microbe can hydrolyze and degrade the caffeine content in coffee cherry during the fermentation. Tawali 2018 reported that caffeine can derive, become chlorogenic acid, and continuing degradation to form smaller compound that can release and more soluble in the water. And for the antioxidant activity, we can see that all fermentation methods also did not affect significantly. Fermentation of coffee cherry in 3 conditions or 3 methods resulted cascara which were stable in its antioxidant activity, but the higher is from a spontaneous fermentation, and the second is Lactobacillus plantarum, and the third or the lowest is Saccharomyces cerevisiae. And for the sensory acceptability, we can see that overall the cascara extract from Saccharomyces cerevisiae fermentation obtain the highest value of acceptability. This indicates that the fermentation process using Saccharomyces cerevisiae can improve the taste quality of cascara beverage or cascara extract than using Lactobacillus plantarum and spontaneous fermentation. For conclusion, the first fermentation processing method of Arabica coffee cherry influence the phenolic, flavonoid, caffeine content, and sensory acceptability. And for Lactobacillus plantarum fermentation, so the highest polyphenol content, caffeine content, and the spontaneous fermentation resulted the highest flavonoid content and antioxidant activity. And for the sensory evaluation, showed that cascara from yeast fermented cherry obtained the highest overall acceptance score of 4 out of 6. And we suggest that the fermentation method of coffee cherry continuous development to optimize the fermentation condition to improve the coffee bean and cascara quality. And thank you for the opportunity, and wassalamualaikum warahmatullahi wabarakatuh.
Thank you to Miss Annie for your presentation. And we will continue to the next presenter is Dian Musdalifa from Research Center for Chemistry LIPI with the title Effect of Soaking Medium on the Nutrients and Hypoglycemic Property of Pre-Cooked Red Kidney Beans. To the co-host, could you please share the next video of the presenter? Thank you.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. My name is Dian Musdalifa. Today I will present my research work entitled Effect of Soaking Medium on the Nutrient and Hypoglycemic Property of Pre-Cooked Red Bean. As we know, red bean is one of the nutritious legumes that possess health benefits including lowering cholesterol level, preventing colon cancer, controlling blood glucose, and maintaining normal body weight. The benefits of consuming red bean are greatly influenced by its nutritional content and active component. The previous study reported that red bean has amount of resistant starch causing a low glycemic index and hypoglycemic property. Like another starchy food, red bean should be cooked before consumption to get the right texture and flavor. The cooking process of red bean that usually takes 8 to 12 hours is presumed to decrease the interest of modern society to consume the red bean. Therefore, a processing technique is needed to produce the ready-to-cook red bean with desirable organoleptic property. One of the processing techniques that can be applied is the pre-cook technique. Pre-cook is the most commonly starchy food processing technique that apply to improve organoleptic quality and reduce the cooking time of a ready-to-cook product. This technique includes a series of steps where the soaking is one of the critical steps that affect to the quality of the pre-cooked product. Soaking can be done in tap water as well as soaking media containing chemicals such as sodium carbonate, sodium bicarbonate, or ammonium carbonate. However, many previous studies reported that soaking in a chemical containing medium was more effective to reduce the cooking time than soaking in the tap water. Every food processing, a process from a raw material into a ready-to-eat form, not only affect the appearance but also affect the nutritional composition of the product. Previous study reported that different types of soaking media and processing method have a very significant effect on the nutritional composition of red bean, especially its resistant starch content. Changes in a resistant starch content due to processing are presumed to be affected to the hypoglycemic property of the red bean. Therefore, this study aims to determine the effect of soaking medium on the nutrient of pre-cooked red bean. And also, this study aims to determine the effect of soaking medium on the hypoglycemic property of pre-cooked red bean. The study was conducted in 3 stages. The first stage is dedicated to define the optimal process condition to produce the pre-cooked red bean. In the soaking step, the red bean was soaked in different soaking media. There is tap water, sodium carbonate, sodium bicarbonate, and ammonium carbonate with a concentration 4.2% weight per volume. And the soaking process was conducted in room temperature. After the soaking process, the bean was followed by cooking in autoclave and cooling in the after cooking, the bean was cooling in the room temperature for 2 hours and followed by drying at 60 degrees Celsius for 90 hours. Sorry, I mean for 80 hours. The second step is conducted to determine the nutrient composition and preference level of the pre-cooked red kidney bean. The preference level was determined using approximately 170 untrained panelists aged 30 to 50 years. And the last stage is conducted to determine the hypoglycemic property of the pre-cooked red bean using Sprague-Dawley as the animal model. All the obtained data were statistically analyzed using ANOVA, followed by Duncan multiple rate test at a 95% confidence level. The proximate analysis of the bean were showed that the type of soaking media affected to the pre-cooked red bean nutritional composition, especially its resistant starch. As can be seen on this table, that the resistant starch of pre-cooked red bean is higher than raw beans. However, soaking in ammonium carbonate produced pre-cooked red bean with the highest resistance starch content compared to the other bean that soaked in other soaking media. The effect of soaking media on the resistance starch of bean is suspected to be related to the pH of the soaking media. The pH of tap water and ammonium carbonate is in the range of natural pH, while the sodium carbonate and sodium bicarbonate the pH is in the range of the basic pH. Beans that soak in a media with basic pH have a lower resistance starch than other beans that soak in a media with a neutral pH. It is expected that alkaline condition cause the structure of starch becoming less compact, leading a reducing of the tendency for retrogradation process. Based on the panel preference test, the type of soaking media affected the preference of the panelists into the pre-cooked red beans. Overall, the panelist preference of the pre-cooked red bean ranged from 4.32, that is neutral to moderate light, to 5.70, that is moderate light to light. The highest preference level was obtained by pre-cooked beans that soak in ammonium carbonate, and the lowest one was obtained by pre-cooked beans that soak in a sodium carbonate. Therefore, the pre-cooked red beans that soak in ammonium carbonate will be tested further for its hypoglycemic property in experimental animal model. The hypoglycemic property assay using animal model were showed that the pre-cooking process affects the hypoglycemic property of red bean, indicated by a decrease in glucoserum level in experimental animal model that have been injected with alloxan on the neat day and fed by the pre-cooked beans. As can be seen in this slide, the experimental animal group that was fed by standard diet containing processing bean was able to reduce the glucoserum level in the range of 43.65 to 55.58% at the end of the test period. Compared to the group that fed by a standard diet. However, the highest reduction in glucoserum level was obtained by the 36.90% of resistant starch, while boiled the beans only contain 30.04%. As a conclusion, the soaking media type had effect on the nutrient composition and hypoglycemic property of pre-cooked red bean. Compared to the tap water soaking process, the usage of soaking medium produced more nutritious and better hypoglycemic property of pre-cooked red bean. And this is the end of my presentation. Thank you very much, and wassalamualaikum warahmatullahi wabarakatuh.
Thank you to Ms. Dian Musdalifa for the nice presentation. And we will move to the next presenter. The next presenter is Ms. Emma Damayanti from Research Unit for Natural Product Technology LIPI. The title of the presentation is antifungal activities of lactic acid bacteria and yeast isolated from various types of tempe.
Indonesian Institute of Sciences, Yogyakarta, Indonesia, and from Institut Teknologi 10 November, Surabaya. Our research about antifungal activities of lactic acid bacteria and yeast isolated from various types of tempe. For introduction, mold versus lactic acid bacteria and yeast. This figure about mycotoxin producing mold like Aspergillus, Fusarium, and Penicillium. This microbe produce mycotoxins such as aflatoxin, ochratoxin, and patulin. This microbe, known as a contaminant in grain-based food, and if this contaminant consumed by human, it can cause heart problem because this mycotoxin as effect as carcinogen, mutagen, teratogen, and immunosuppressant. In the other hand, lactic acid bacteria and yeast can inhibit mycotoxin producing mold and inhibit aflatoxin production. This microbe is common found in fermented product. Indonesia has a large variety of traditional fermented food, and tempe is the famous one as a fermented soybean. Other type of traditional tempe like tempe lam toro made from betai Cina bean and tempe koro made from sword bean or koro pedang, and then tempe gembus and oncom made from byproduct include peanut, soybean curd, tapioca byproduct or onggok. This figure is about 4 type of tempe used in this research. Lactic acid bacteria and yeast reported as tempe microbiota, but lactic acid bacteria and yeast associated with various type of tempe has not been widely reported. The objective of this research, we will isolate lactic acid bacteria and yeast associated with various type of tempe and to select lactic acid bacteria and yeast isolate which have antifungal activity against mycotoxin producing mold. Material and method, tempe sample obtained from individual producer in Gading Village, Gunung Kidul, and in traditional market in Bantul, Yogyakarta. For yeast and lactic acid bacteria enumeration, we use the main rugosa sharp agar, MRSA, for lactic acid bacteria, and chloramphenicol yeast glucose agar for yeast. Universitas Gadjah Mada. This mode is known as a mycotoxin producing mold. Parasiticus and citrinum grown on mea medium, malt extract agar medium, and incubate for 5 until 7 days until produce the spores, and spore harvest by aid twin egg and homogenized and taken aseptically, and then mold spore concentration calculated and adjust to 10 million per milli. For antifungal assay, we use 2 methods, qualitative and quantitative. And in qualitative antifungal, we use a strict plate method in selected medium, and then we use soft meal, malt extract agar contain mold spore, added in the plate, and then after incubation in 2 until 3 days, the region around the lactic acid bacteria and yeast colony indicate antifungal activity, and we can show in this figure. This is about antifungal activity. And in quantitative antifungal assay, we use this distribution method. We use supernatant containing secondary metabolite of lactic acid bacteria and yeast, and drip on this paper and place on the surface media containing mold spot. inhibition zone diameter measured after incubation in 3 days. This figure shows that all various type of tempe sample and various type of mold and yeast grow on agar medium. In nutrition and aflatoxin content of tempe, we evaluate carbohydrate, crude protein, fat, and crude fiber. This all tempe has a variety of this parameter. But in aflatoxin content, all tempe has a content aflatoxin under 5 ppb, and in regulation, the maximum limit of total aflatoxin in food or feed product from Badan POM RI has 35 ppb, and US FDA, Food Drug Administration, 20 ppb, and European Public Health Division 4 until 15 ppb. And we assume that this product safe product for consumption. And we predict that the low aflatoxin content in all tempe is possible due to the presence of lactic acid bacteria and yeast, like in this figure. This figure 2 show that lactic acid bacteria and yeast population has a variety in the number. The highest lactic acid bacteria found in oncom and the highest number of yeast found in tempe gembus. The variation in the number of lactic acid bacteria caused by various on substrate, mold type, and process in each type of tempe. In antifungal activity, using qualitative and quantitative method, we found lactic acid bacteria G1 and yeast GCK1 and GCK2, all microbe isolated from tempe gembus, has the highest average inhibition zone. This the morphological characteristic of Saccharomyces in figure B1 and lactic acid bacteria Lactobacillus in B2. This is a specific characteristic for Saccharomyces and Lactobacillus. For conclusion, the total aflatoxin in all tempe product under 5 ppb and qualified as food consumption. The total lactic acid bacteria in tempe product higher than total yeast, and then Lactobacillus G1 and Saccharomyces spp GCK1 and GCK2 isolated from tempe gembus has the highest antifungal activity against mycotoxin producing fungi like Aspergillus parasiticus and Penicillium citrinum. The selected lactic acid bacteria and yeast were potential to be used as natural biopreservative in functional food product to prevent the growth of mycotoxin producing fungi. This research was funded by DIPA BPTB LIPI 2014, and we thank Sri Endatini and Madina Roma and Lusti Istiqomah for technical assistance in aflatoxin analysis. Thank you very much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you to Ms. Emma for your presentation. And now we have come to the last presenter for this session. The presentation will be delivered by Ms. Novita Indrianti from Research Center for Appropriate Technology in LIPI with the title Pumpkin Flour Effects on Antioxidant Activity, Texture, and Sensory Attributes of Flat Tubers Noodle. For the co-host, please proceed for the last video. Thank you.
Good afternoon. My name is Novita Indrianti. Let me present our research entitled Pumpkin Flour Effects on Antioxidant Activity, Texture, and Sensory Attributes of Flat Tuber Noodles. As we know, noodles are the most popular Indonesian food. In general, noodle made from wheat flour, but in non-wheat noodles, the absence of wheat flour can be replaced by using flour or starch from tubers. Flat noodles are one type of noodles which characterized with a thin flat shape and is generally made from starch. The texture of noodle is important parameter affecting noodle quality. The height stickiness is an undesirable texture for noodle quality. Kana and arrowroot are local tubers which have a high amylose content. The high amylose content of this starch resulting low stickiness and more compact of noodles. Color is one of a critical parameter that indicated visual appearance and overall acceptability of noodles. The color of noodle can be improved by adding natural ingredients to the noodle. Pumpkin is one of the most desirable vegetables because it has yellow color and rich in carotenoid and that can act antioxidant. In our study, we made flat noodles from a mixture of tuber starch and pumpkin flour. The purpose of our research was studied to the effect of pumpkin flour on texture, antioxidant activity, and sensory attributes of flat tuber noodles. My next point is about the method of this research. The ingredients were used for making flat tuber noodle are kana starch, arrowroot starch, pumpkin flour, salt, and water. The treatments which were carried out in the study were different level concentration of pumpkin flour included 5%, 10%, and 15% of pumpkin flour. The flat tuber noodles were prepared by mixing kana or arrowroot starch with pumpkin flour, salt, and water. The slurry poured into an aluminum tray and steamed for 3 minutes. The flat tuber noodle seed dried an oven dryer at 50 degrees Celsius to half dry. The half dry seed cut off into small seeds and dried again. The flat tuber noodles were analyzed antioxidant pH method. Texture was analyzed using texture analyzer and sensory attributes included color, aroma, taste, texture, and overall acceptability were evaluated by 30 untrained panelists using 7-point hedonic scale. The next area I want to discuss is the results of our research. From figure 1, it could be observed that the addition of pumpkin flour increased the antioxidant activity of flat tuber noodles compared to control. The results can be due to the fact that pumpkins are rich in carotenoid, which function as an antioxidant. The texture of a noodle was expressed with the stickiness. The stickiness of flat tuber noodles can be seen on figure 2. The addition of pumpkin flour determined an increase in the stickiness of flat tuber noodles. The addition of pumpkin flour increased the water absorption of milk flour, which resulted in noodle more sticky. The stickiness of flat kana noodles was lower than the stickiness of flat arrowroot noodles. According to the result of the noodle stickiness, 3 flat tuber noodles with the lowest stickiness included a flat canna noodle with pumpkin flour 5%, 10%, and 50% were evaluated the sensory attributes. From figure 3, we can conclude that the aroma and taste of flat noodles did not vary with increasing of pumpkin flour's level. Flat canna noodle with 10% of pumpkin flour obtained the highest score for color, texture, and overall acceptability. The conclusion of our research are the addition of pumpkin flour increased antioxidant activity and the thickness of flat tuber's noodle. Flat canna noodle with 10% of pumpkin flour was the most favored by panelists. Antioxidant activity and the stickiness of flat canna noodle with 10% of pumpkin flour were 39.80% and 3,350 gram force respectively. Okay, this is the last of my presentation, and thank you.
Okay, so before we continue to the discussion session, I would like to thank for all the presenter for their great presentation and deliver about and sharing their research project, and hopefully we can have widen our knowledge and also have a fruitful discussion. So we will continue to the discussion session. We already have some questions to the presenter. So to the first presenter, Miss Annie Soliha, we have two questions. First from Miss Novita Indrianti. What is the meaning of Z, Y, X, Y, Z in your result? Could you explain more in detail? And the second question from Miss Emma Damayanti is how the mechanism of lactobacillus could increase the caffeine content. Is there a specific enzyme that plays a role in converting caffeine? To Miss Annie Soliha, you may answer the question from the audience. Thank you.
Okay, thank you, Ms. Hakiki. Okay, thank you for Ms. Novita Indiati and Ms. Emma for your question. The first, I'm sorry because maybe to write in my slide or in my presentation about the meaning of XYZ, but in our full paper or full manuscript, I already write it completely. Okay, for X is spontaneous fermentation, and the Y.
Sorry.
Okay.
Sorry, Miss Annie. Maybe you can also share screen of your PPT file.
Okay.
So it will be more clearer.
Okay. Could you see my slide?
The slide is already seen.
Okay. Sorry, this is the meaning of X, Y, and Z, which is the X is for spontaneous fermentation, and Y for Lactobacillus plantarum, and Z for fermentation using Saccharomyces cerevisiae. And I think it's clear about the sample.
Yes.
Okay. And the next about the caffeine content. for Miss Emma. Thank you, Mbak Emma, for your question. About the caffeine content in this cascara or in the infused water of cascara, we can see that the spontaneous fermentation has the lowest value or the lowest content of caffeine, and for lactobacillus plantarum, microbe hydrolyze the compound that conjugate with caffeine, so it's makes or cause the caffeine release to the water. So in the spontaneous fermentation, the caffeine content is the lowest level, but in the fermentation using only lactobacillus plantarum, we obtain the highest level, but it's not mean I don't know. Maybe Bu Dewi can help me to explain more, maybe, but from my knowledge, that's it. I just it, Mbak Emma. Thank you.
Thank you to Miss Annie for answering the question. I think it's already clear. For the second question to Miss Dian is from Novita Indrianti. How is the mechanism medium soaking can improve nutrient quality, especially in resistant starch? So to Miss Dian Musdalifa, please, time is yours. You may answer the questions.
Okay, thank you. Thank you for the question. I have tried to answer. First, for the first question is from Mr. Ahmad. Yeah, you asked about what kind of enzyme did you use. Generally, for the resistance start analysis, I use the Kuni et al. Method. If you need the paper, I can send you directly, privately by email. But here, I can notice that in this analysis, we use pepsin, alpha amylase, and amyloglucosidase. And then, next question, did you do fat removal prior to the analysis? No, I don't remove the fat because this analysis won't to gastrointestinal digestion. So if we remove the fat prior to analysis, I think it's not similar with the gastrointestinal digestion. And then, how long enzyme digestion did you do? For pepsin digestion, it's conduct for 60 minutes, and then for alpha amylase digestion is conduct for 16 hours, and then for amyloglucosidase incubation, it's conduct for 45 minutes. I think it's clear enough. And then I will move to the next question from Novita Indrianti. It's about how is the mechanism medium soaking can improve nutrient quality, especially in resistant starch. If I'm not mistaken, I mentioned before in the presentation that the resistant starch of bean is suspected to be related to the pH of the soaking media. As I said before, that this soaking media is tap water, sodium carbonate, sodium bicarbonate, and ammonium carbonate. Tap water and the ammonium carbonate have neutral pH. However, sodium carbonate and sodium bicarbonate have the alkaline pH. The tendency is when the beans are soaking in the alkaline pH medium, the resistant starch is lower than the bean which soaked in the natural pH. So it's expected that alkaline condition cause the structure of the starch becoming less compact, leading a reducing of the tendency for the starch going to retrogradation process. Less compact because I expected that the sodium Na plus will be make a bridge between the amylose chain, so it will inhibit the retrogradation between amylose to be a.
Okay, so we will have next question for Miss Emma Damayanti from Mr. Ahmad. Is there any way to remove aflatoxin from tempe so that its content in tempe is 0?
Okay.
To Miss Emma Damayanti, time is yours.
Thank you for the question from Mr. Ahmad. I'm sorry, maybe I'm not sure if there is a treatment to remove aflatoxin in tempe until 0 content, because as we know, in European Public Health Division, the limit of aflatoxin content in food 15 ppb. So 15 ppb is the highest and the limit for consumption. And in Indonesia, only 35 and tempe sample in this study, the aflatoxin content under 5 for consumption, and we can assume that is safe for food consumption in all type of tempe. Sorry, maybe I am not sure if the aflatoxin content became 0 in food from bean, because the aflatoxin is like a natural. Our country is a tropical country, and so the humidity, environment, and a lot of variety of microbe such as contaminant everywhere and every time can contaminant as a contaminant in food. Okay, maybe it's clear. Thank you.
Okay, thank you, Miss Damayanti. Is it clear for Mr. Ahmad? Okay, is there any question for the present presenter or the previous presenter? Because maybe we still have time for around 1 or 2 questions. If you have question, maybe you can also ask directly in this forum. Maybe I would like to ask to the Miss Novita Indrianti, the last presenter. So in the presentation, you mentioned about the pumpkin flour that has been applied to the flat tubers noodle. Is there any other possible application that we can use by using this pumpkin flour? Maybe you can explain another application other than flat tubers noodle. Thank you.
Thank you for the question. I think the application of pumpkin flour is very can apply for bakery product or pasta product. I think the application of pumpkin flour is very luas, sangat luas ya. It's very widely.
Okay, thank you. So it also has a potential to be applied to another food products, maybe such as cookies or biscuit or something else. Okay,
Yes.
thank you for your answer. And we also have another question from Miss Lusty to Miss Annie Soliha. If the Lactobacillus plantarum produce the highest polyphenol while Saccharomyces cerevisiae reveal the good sensory, then whether we could combine both microbes as multiculture inoculum for fermentation to obtain the targeted properties? To Miss Annie Soliha,
Okay.
please answer the
Okay,
question.
thank you to Miss Lusti. I think I agree with you that we can continue this research about to combine group to produce the more better quality for the cascara or for the coffee bean that we will obtain from this fermentation process. Okay, thank you.
But are you do you plan in the future that you will use this multiculture for your next research? Do you have any plan to use this multiculture for your next research?
I hope we can collaborate because I think this researcher in this room or in this conference have competence in fermentation or microbiological technology. Maybe we can collaborate to the food processing technology based on the microbial, especially for cascara or for coffee processing. Thank you.
Okay, thank you, Miss Annie Soliha. So we also hope through this event we can also open such research collaboration with other institutions, as many as we can. And I think we also already finished this session. So once again, I would like to thank all the presenters and all the participants that has been join this parallel session. Even though we cannot meet physically, but virtually, but even though I hope we still can benefit many from this conference. Thank you very much. And then we will have a break time until 3:15, and hope that you can join back to the next parallel session. Okay. Thank you. Thank you very much for all of your attention and have a nice day. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Secretary and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation, significant and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research. Scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers. 2. Accrediting scientific journals. 3. Studying and evaluating biodiversity. Establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO and has 3 international centers, namely Asia Pacific Centers for Eco-Hydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future. Each vision begins with a spark of inspiration, and each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research. Consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism.
Okay, well, everybody, this is time for us to start our program again. Okay, good evening, all distinguished participants. Assalamualaikum warahmatullahi wabarakatuh.
Warahmatullahi wabarakatuh.
Welcome back to our program. I hope all of you still on the good and fresh condition. So this evening, we have the second parallel session on room C. I'm Lisa. Here I'm going to assist you as a moderator. So before we start our parallel session, let me repeat again about the rule for the participants. To all participants, please be advised that the chat room is only for 1. Delivering question to the ongoing discussion in the session. 2. Requesting for changing or moving to the other breakout room in the parallel session. And 3. Announcements delivered by the organizing committees. Don't forget to fill in the registration form in the link on the chat room for the session 2. And for question and answer in this parallel session, please write down in the chat room with a format from your name underscores your institution to panelist name who you want to ask the question. Or you can raise your hand to ask directly during discussion session. And for all of the participants, please properly rename your Zoom ID according to your participant category to set you up to the correct breakout room in the parallel session. Okay, So in this session, we have 6 presenters. As the committee already announced before to the participants that each presenter has 10 minutes time to deliver their presentation and 5 minutes for discussion. So before we start the session, I would like to divide the presenters today into 2 groups. So the top 3 presenters, it means C21, C22, and C23 would deliver this presentation first and followed by discussion session. After discussion time, we continue to second group, C24, C25, and C26 that would deliver their presentation and close by discussion session. Okay, for the first presenter, I would like to invite Ms. Agustin from Indonesian Institute of Sciences. She would like to deliver her presentation with the title of Alternative Use of Modified Cassava Flour, MOCAF, as Carrier of Natural Folic Acid in Infant Biscuit for Complementary Feeding. Please, for the Mr. Asa, co-host, please prepare for her video. Time is yours.
Mocha flour for carrier folic acid and creepiness on biscuit for complementary feeding. The aim of experimental activity just for know best mocha concentration as carrier reducing sugar, folic acid, this is important, and for characteristic of biscuit, we using LCMS for know about folic acid monomer and particle size distribution, volatile compound by GCMS, and lightness for biscuit. This is about how we on preparing biscuit for complementary feeding. First, we get to fortificant of natural folic acid, 1 part broccoli, fermented broccoli, and 1 part soy or mung bean tempeh, mixing, and then we adding with nixtama, yellow corn, 1 part, and homogenizing, then drying. We get a fortificant of natural folic acid. For get a biscuit, we on preparing by mixing of formulation with biscuit formula and then molding and baking. We get the infant biscuit as complementary feeding. The results of research, I show you how about characteristic of material. This is all material for using in this research. The kind of material different about composition, especially of folic acid, fortificant A, B, and this formula and mocha. You see how about different concentration. This is effect of formulation on composition biscuit. I show you how influencing concentration of mocha for folic acid on biscuit. Biscuit A we using on fortificant A and biscuit B we using on fortificant B. The result optimum using mocha show how base of mocha concentration at 0.3%, but for dissolved protein it's different. Using fortificant A, and this is effect for total solid, total sugar, and reducing sugar. So I want to show you how combination of optimum process of infant biscuit. The optimum process using of fortifican B and fortifican A, who fortifican A on concentration 0.3%, but when using fortifican B on concentration mock-up to 0.4%. I show you how the photo of infant biscuit A and B and how composition. There are many different composition, especially for folic acid. Infant biscuit B show how folic acid is better than infant biscuit A. How about identification of folic acid monomer? This is just for identification of monomer for molecule, basic of molecule weight, who on biscuit A dominant of monomer folic acid. It's different. You see how on infant biscuit B using we get dominated 3 monomer with molecule weight on different. How about distribution of particle size? You can look how infant biscuit B, it's for average more big, more large than particle size of biscuit A. How about identification of volatile compound? We know we using mocha who we got for mocha by fermentation of lactobacillus bacteria. So when you look how about compound, it's different. Infant biscuit B get acetic acid more small than infant biscuit A, but another compound it's same. How identification of crispiness and sharpness of biscuit? This picture show you how lightness for biscuit A more lightness than biscuit B. How conclusion? Based on folic acid recover, optimization treatment get a mock-up concentration on 0.3%, and for using fortifican B it's a same concentration mock-up same. In this optimization formulation, using fortifican B, increase folic acid twofold when compared with its component in this formula. And infant biscuit B were giving characteristic of dominant folic acid, 3 monomer, and a particle size of rich like we saw in this presentation. Thanks so much for funding by Ministry of Research, LIPI, and very thanks for Indonesian Institute of Science too. Thanks so much for your attention. Please don't ask too much.
Okay, thank you, Ms. Agustin. That was very nice presentation. So, well, we go to the second presenter. I would like to invite Miss Uyeria Andayani. Since we don't have her video already, sorry. Since we don't have her video already, so maybe Miss Uyeria want to present by live. So Miss Uyeria, are you here? May I call again? Miss Uyeria Andayani. Are you joining the session? Are you joining the session? Miss Uyeria? Okay, I think Miss Uyeria doesn't come today. So we can continue to the third presenter. So I would like to invite the third presenter, Miss Kristanti from Indonesian Institute of Sciences. She would like to deliver the presentation with the title Physicochemical and Microbiological Properties of Pudding Powder as a Complementary Food During Storage. So Miss Kristanti, time is yours. Please, Mr. Asa, prepare her video.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Hello, ladies and gentlemen. I would like to introduce myself. My name is Tita Kristanti and my partner is Dr. Ainia Herminiati. We are researchers from Research Center for Appropriate Technology, Indonesian Institute of Sciences. In this section, I would like to present our research, physical, chemical, and microbiological properties of pudding powder as a complementary food during storage. We have developed the inulin-enriched pudding powder for complementary food to support the growth and development of children over 1 year old. In 2018, we developed the inulin-enriched pudding for complementary food that got good responses from both mother and children under 5 years old by the sensory evaluation. However, the iron and zinc contents did not meet the SNI requirement for instant complementary food powder. And then in 2019, we fortified the inulin-enriched pudding powder with micronutrient premix contains iron and zinc. The iron and zinc content has increased by tripled, and the zinc content of the pudding powder did meet the SNI requirement for instant complementary food powder. The complementary food must be safe for infants, so evaluation of the physical, chemical, and microbiological properties during storage is most important. It related to food safety and customer acceptance. According to the background, this research aims to evaluate the physical, chemical, and microbiological properties of pudding powder during storage. The material used in this research were skim milk powder, modified cassava flour or mocav, sugar powder, carrageenan, inulin, and micronutrient premix. The pudding powder was prepared according to Kristanti and Herminiati 2019, and then the pudding powder were packaged in polyethylene plastic and aluminum foil pouches, then stored at 15 degrees Celsius, 30 degrees Celsius, and 45 degrees Celsius for 4 weeks. The pudding powder were analyzed every week during storage time. The analyzed were moisture content, water activity, weight of pudding powder, protein, color properties, and total plate count or TPC. The data were analyzed using IBM SPSS statistic 20 and analysis of variance with Duncan means test. Result and discussion. The moisture content of pudding powder were significantly decreased during stores in 4 weeks, except for PE30, which was increased. The PE45 sample had a highest decline in moisture content, and it formed an agglomerate. The mushroom research showed that the storage of instant milk formula powder at 40 degrees Celsius for 15 days caused very loss of moisture contents and powder was caked. The factor may contribute to moist content during storage are permeability of packaging, storage temperature, and storage time. The weight of pudding powder during storage. The weight of the pudding powder storage in aluminum foil were relatively stable after first week storage. Furthermore, the weight of pudding storage in polyethylene plastic were significantly increased during storage, except for the PE45 sample, which was drastically declined. The increasing of the PE15 and PE30 may be caused by the increasing of moisture content during storage. And then the drastically decline of the PE45 sample was due to drastically decrease of moisture content during storage. The water activity of pudding powder during storage. The water activity of PE15, PE13, and PF45 significantly increased after storage. However, the water activity of PE45, EF15, and EF13 were significantly decreased after storage. The increasing of the water activity of EF45 was the highest among others. The change in water activity of the pudding powder are caused by the change in moisture content during the storage period. It's according to the Yu et al. Study that the increase of water activity was accompanied by the increase of the moisture content. The permeability of the packaging affect the characteristic of the food product during storage, especially moisture content and water activity. Generally, the high packaging permeability makes the characteristic of the food product more easier to change, and then so the shelf life is short. Protein content pudding powder during storage. The protein is the one of the parameter which can be affected by the temperature change during storage. The highest decline of the protein after 4-week storage occurred in the EF45 sample. The decreasing in the protein content in the pudding powder is probably caused by the protein denaturation during storage. The water activity of the pudding powder also may play on the damaged protein. And then, color properties of pudding powder during storage. The lightness of the pudding powder was significantly decreased all sample. The F value of the PE pouch sample increased while the EF pouch decreased, but the value were not significantly different. B value of PE15, PE30, and EF15 significantly decreased, but the EF30 and EF45 was increased. The EF45 was lowest of lightness and highest of B value, was darkest and yellowness colored, and formed an agglomerate. The storage caused the increasing of the color different B value and Maillard browning index value in the high protein product. The factor effect of the likeness of the product during storage are type of the packaging, storage temperature, sugar, and protein content of product, water activity, and storage time. The total plate count or TPC in all pudding powder samples significantly increased after 4-week storage. The TPC of the pudding powder storage at the 15 degrees Celsius and 30 degrees Celsius in both polyethylene and aluminum foil 3rd-week storage. These results were consistent with the previous study. The TPC of the instant porridge as complementary food increased during storage in plastic bag at 15 degrees Celsius for 4 weeks. And then another research showed that the TPC of the mango milk powder decreased during storage in the tin can, metallized polyester, and polyestyrene bag at 5 degrees Celsius and 15 degrees Celsius for 8 months. The increasing of the TPC during storage is probably caused by the hike of the water activity in pudding powder. The high value of the water activity affect the probability of microbial growth, result in the increasing of the total bacteria in pudding powder. Conclusion: the study and aluminum foil pouches were at temperature 15 degrees Celsius. Furthermore, it is necessary to conduct sensory evaluation during storage in order to determine the consumer acceptance level. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Ms. Kristanti, for the nice presentation. So, because Ms. Wuyeria doesn't come today, I would like to continue to the fourth presenter. I would like to invite Ms. Kamnatania from Universitas Pelita Harapan. She will deliver the presentation with the title Bitterness Reduction of Green Cirrheta Andrographis pandiculata leaves and its functionality. So, for the co-host, please prepare the video, and Ms. Natania, time is yours.
Good afternoon to all the ISAC committee, to the moderator, and to all the participants. Thank you for this opportunity to present some of the results from my research entitled Bitterness Reduction of Green Cirrheta leaf and its Functionality. My name is Natania. I currently work as lecturer in UPH in Food Technology Department. These are several of my students. Some of them are working together in this research project. Some of them are already graduated by now. In this research, we focus on the bitter herbs of Sambiloto. Sambiloto is a traditional herb known for their functionality. It has many notable antioxidant activity, anti-inflammatory, anti-diabetic, anti-cancer, and many other functional benefits. However, Sambiloto itself is very infamous for their bitter taste. If you have ever tried the Sambiloto, you will never forget their distinct bitter taste. So in this research, what we want to do is we try to reduce the bitterness of the Sambiloto. We derive our idea from the usage of clay or tanah lempung. In rural area, women are used the clay or tanah lempung to reduce or to remove the bitterness of various herbal plants. Throughout our journal study, we also found that the application of adsorbent is actually can remove the bitter component, limonene, from orange juice up to 96% in the presence of temperate heat. In this research, we use a slight modern approach. We do not use the traditional clay because the clay itself consists of very different materials and it differs from place to place. So we try to use the semi-purified type of clay, which is bentonite clay, activated atapulgite, and diatomaceous earth. These three types of clay, semi-purified clay, are silica-based, but they all different in the type of minerals attached to, thus making these three type of absorbent have different absorbing features. However, the removal of bitterness, though the bitterness is undesirable, it's closely related to the presence of phytochemicals or functional properties. And the removal of bitterness might cause that the Sambiloto itself will lose its functional property. Therefore, what we do in our research project is we observe how these three different absorbents, which is diatomite, bentonite, and activated atapulgite, and the application of different stage of heating, 5 minutes, 10 minutes, and 20 minutes boiling, in their effectivity to remove the bitterness and how it might affect their functional and also the phytochemical content. In the sensory analysis, we use scalar tests to calculate the intensity of the bitterness, we actually screened several panelists. We started from around 50 panelists, and we end up after final elimination around 12 panelists. We choose them based on their ability to differentiate taste and also their ability to discern the level of the bitter taste. And at the end of the selection, we train them to have the same perception in scaling the bitterness level of different bitterness intensity to make sure that we can calculate the bitterness correctly. For the phytochemical analysis, the one that we analyze is the total terpenoid content, total tannin, alkaloid are able to reduce the bitterness of the Sambiloto leaf. If you see from the scale 1 to 15, the application of atapulgit can reduce the bitterness of Sambiloto starting from 14 up to 1.37. So at most, atapulgit is able to remove around 90% of the bitterness of the Sambiloto. The increasing heat, the increasing time of heating also increase the amount of bitterness removal in the Sambiloto itself. As for the phytochemical contents, we could see also several trends. The similar trend is the increase of heating, resulting in the increase of removal of the phytochemical compound. So the longer the heating period, the more phytochemical is lost during treatment, after treatment. And for terpenoid content, we could see that the activated atapulgit is the one that is able to reduce almost 78% of the terpenoid content from the Sambiloto itself. For the tannin content, we see again that the atapulgit can reduce the most of the tannin content. For other phytochemicals such as alkaloid, flavonoid, and phenolic compound, we could see that the bentonite, the application of bentonite is the one that can reduce the most of those three compounds. So they are slightly different between each component and the adsorbent that is more effective in removing the component. All of this result is quite alarming actually, because we might end up with the Sambiloto or the herbs without functional property. We all have 454 IC50 ppm for the IC50, and the atapulgit still have around 680 ppm. So it's actually still quite good. It only slightly reduces the antioxidant activity of the green chirata leaves. To further observe the effect of bitterness and also the bitterness removal and the content, we use Pearson correlation to analyze the data, and we could found that bitterness is actually closely related to the terpenoid content, while the IC50 is closely related to the alkaloid, phenolic, and flavonoid. However, interestingly, we can find that there is no significant correlation between antioxidant activity and bitterness. These are exciting findings. For this mean we could reduce the bitterness of the Sambiloto without much affecting their functional properties. So the conclusion of this research are the application of adsorbent and increased boiling time can reduce the bitterness and functional property of green chirata leaf. With atapulgit, accompanied with 20 minutes boiling time, was able to reduce the most of bitterness of the leaves. However, we found out that there is no correlation between bitterness reduction and antioxidant activity. Thus, the application of adsorbent has a potential to be used to increase the palatability of the green chirata. Thank you for your attention. If you have any question, I will be happy to answer the question. Thank you.
Okay, thank you, Ms. Nathania, for the nice presentation. So this is the end of the first group for our parallel session. Now we go to the discussion session, and I would remind you, you can write down your question on the chat room. Now we have one question on the chat room from Miss Lusti Istiqomah to Miss Dita Kristanti. The question is, if we compare to the storage condition of commercial pudding powder at room temperature, while in this study the product storage at 15 degrees Celsius. Could you explain what material affect the different result? So please, Miss Dita, can you
Okay.
answer the question?
Okay, thank you, moderator. And the pudding powder in our research content of sugar and skim milk powder that is hygroscopic. So the moisture content is higher, and then its effect on the life storage of the pudding powder in our research. And then the difference with material in our research with the commercial is that the commercial pudding usually addition of the antioxidant, so the its impact on the life storage that long term life storage than my our pudding powder. Okay, this is my answer.
Thank you. Is there any question from Miss Lusti? Is it okay? Is it clear?
What about the methodology? Is it different with the commercial process?
Can you repeat again? Sorry.
What about the methodology to process the pudding powder? Is it same or different with the commercial pudding?
Okay. I think it's different because in this research we just use the mixer for the drying, mixing for the ingredient of the pudding powder, and then at the commercial maybe use the spray dry or drum dry, etc. Okay, thank you. Is this enough? Clear?
Yes, enough.
Thank you.
Okay, thank you, Miss Dita. So we have another question from Miss Emma Damayanti to Miss Natania. How is the mechanism of bentonite binding to bitterness properties in Sambiloto? Please, Miss Natania, can answer.
Okay, thank you for the question. Sorry. The bentonite itself, they have the what they call the cation capacity, surface capacity. So it's actually the outer part of the bentonite. They have several cation, and it actually can bind with the phytochemicals. That's why because of the different types of the adsorbents, so they bind a little bit differently depending on the component of the phytochemical that they want to bind. For the bentonite itself, because they have actually they are silica based, and they have the cation outside those silica, the other minerals, that it will capture some of the phytochemical. That's why it removes the phytochemical from the Sambiloto itself after application with heating.
Okay, Miss Emma, is it clear? Clear enough?
Reduction of bitterness not only from bentonite but also from boiling process.
Actually, if we just use the boiling process, it will not remove the bitterness of the Sambiloto.
Okay.
You need to add the adsorbents, and I think we still don't know exactly the mechanism. It's just guessing that because the adsorbent, they have what they call the cation capacity.
Okay.
So it can attract some of the phytochemicals because the phytochemicals they.
Miss Natania, you still have another question from Mr. Ahmad Sarifuddin. The question is there any side effect of using diatomite bentonite atau pugit to remove bitterness of Sambiloto?
Yeah, so far the side effect is we slightly lose the activity. One of the students already submitted the anti-diabetic result, and the anti-diabetic also they have some reductions in the activity. So it's not much because we use the IC50 methods to analyze the activity for antioxidant and anti-diabetic. We still have the activity of antioxidant, anti-diabetic, but it is reduced. So the application of adsorbent side effect probably will reduce the functionality. However, if you are familiar with the antioxidant property, the value of the removal is still quite low. So extract a good properties, antioxidant property from the extract of Sambiloto. It's actually still have a good property, still considered as a good antioxidant source. However, it's reduced, not as good as the fresh Sambiloto with the bitter taste.
Okay. I think the answer is very well. So in the chat room we don't have any questions. So maybe anybody want to ask some question, you can raise your hand directly. We still have about 8 minutes for our discussion session. You can raise your hand. Okay, Miss Lucy.
Thank you very much for the opportunity. I'm curious about the presentation from Mrs. Natania. Do you also calculate the residual of each absorbent in the final product? And does the residual safe for our human consumption? Thank you.
Okay, we do not check because what we did is we extract the component, the phytochemical component. So we just clean the loto after boiling with the absorbents, and then we extract. So we don't calculate the residual. Yeah, we assume that we component using ethanol, then we don't extract the absorbent, but we check the residuals. If they want to be applied for actual consumptions, we can check the residual.
Okay.
That's the point.
Thank you.
Okay, Missy. Okay. Fine. Maybe this is the end of our first session, part from our session. So we will continue to the second group because I think Miss Uyeria is still not here and nobody represent her. So we still have 2 presenters left. So now I would like to invite the 6 presenters. Miss Ratnawati from Indonesian Sciences. She will deliver the presentation with the title, The Effect of Particle Size and Brewing Time of Ginger Powder to the Characteristic and Acceptance of the Herbal Product. So the time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Thank you, moderator, for the chance. Good evening, everyone. In this good occasion, I would like to present our study with the title product. But before that, let me introduce myself. My name is Ratnawati, and in our team are Dewi Desnilasari, Lista Eka Yulianti, Dita Kristanti, Devri Pramesti Putri, Eni Solihah, Raden Cecep Erwan Andriansyah, and I Research Center for Approach Institute of Sciences, Subang, West Java. As we know that fresh ginger is perishable food and has short shelf life, but it contains active phenolic compounds such as gingerol, sogal, and paradol that have antioxidant, anti-cancer, anti-inflammatory, anti-angiogenesis, and anti-atherosclerotic properties. One of the food processing to improve the quality of ginger is drying to produce ginger powder that have advantage such as good solubility in water, easy to serve, and long shelf life due to its low moisture content. one of the use of ginger powder is an herbal drink, but the study of particle size and brewing time of ginger powder is limited. In the previous study, Makanjuola investigated the influence of particle size and extraction sulfate on antioxidant properties of extract of tea, ginger, and tea ginger blend. Zhao also reported that supervised ginger had many significant characteristics. Sulfate powder was increased, had water holding capacity, water solubility index, and protein solubility, and was well suited to manufacture instant and convenient food. In this study, was to investigate the effect of particle size and brewing time of ginger powder on tons of herbal products. Next, the materials was used in this study TBAC, DPPH, ascorbic acid, ethanol, distilled water, and filter paper. This study conducted using a sign with two factors, particle size, 40, 20, and 10 mass, and brewing time, 5, 10, 15, and 20 minutes. All treatments were carried out in triplicate. Analysis of variance and then contests were performed on the obtained difference. For ginger powder preparation, first, the simplicity of ginger were crushed using dry mill sieve until the ginger powder was obtained. The ginger powder, then analyzed the moisture content. After that, about 3 grams of ginger powder put into tea bag and then brewed according to research design. Then the ginger extract, analyze the extractable matter, color evaluation, antioxidant activity, and sensory evaluation. That requires.
Sorry, I think there is a trouble. I will ask Mr. Asa to fix it. Okay, we can reply, I think. For the last slide, Mr. Asa, could you? Still no sound. I'm sorry for the trouble.
Smaller particle size. This result, in line with the previous studies conducted by Raja et al., state that carica leaf powder with larger particle size also had higher moisture content. Continue to the next slide. We can see from this table that the larger particle size and the longer brewing time, the higher of extractable matter, and the darker the color extract, increase the A value and decrease the B value. Next, we also analyze the antioxidant activity of ginger extract and the data present in this table. From this table, we know that the larger particle size and the longer brewing time can decrease the antioxidant activity. According to Makanjuola, reported that the size reduction of plant substrate before extraction maximize the surface area, which in turn enhance the mass transfer of active compound from plant material to the solvent. In another study, state that the antioxidant activity, total phenol content, and gingerol content of ginger decrease with increasing the time of the thermal processing. The highest antioxidant activity is 20 MES and 5 minutes brewing time, with the antioxidant activity 77.97%. Based on the extractable matter value and the antioxidant activity of ginger extract, the best particularly size for extraction was 20 MES with different brewing time. Analyze the product acceptance through organoleptic tests, including color, aroma test, and overall acceptance. From this table, we choose the best treatment based on higher antioxidant activity and sensory evaluation is ginger extract with 5 minutes brewing time. For the last slide, the conclusion of this study are the larger particle size causes a decrease in the L value, an increase in the E value, and decrease in the B value. The shortest brewing time with 20 MES tends to produce the highest antioxidant activity. In concluding, the ginger powder with 20 MES particle size and 5 minutes brewing time was chosen to be the best treatment based on its antioxidant activity and sensory evaluation. I think enough for my presentation. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. So, now we go to the last presenter. I would like to invite Ms. Indah Kusmartini from National Nuclear Energy Agency of Indonesia, Batan. She will deliver the presentation with the title of Macro and Micro Mineral in Commercial Infant Formula Milk in Indonesia by Neutron Activation Analysis. So, please, Ms. Indah Kusmartini, time is yours. Mr. Asa, please prepare the video.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Thank you for the opportunity to be attending in conference of ISAC 2020. Please allow me to present my paper, and the title of this paper is Macro and Micro Mineral in Commercial Infant Formula Milk in Indonesia by Neutron Activation Analysis. For introduction, as we know, infant milk is a product that requires as a substitute for breast milk that can nutritional supplies of infants from 4 to 6 months of life. The content of formula milk is based on composition of modified breast milk to increase nutritional requirements for dietary intake in infants. Commonly, formula mix comes from cow's milk. However, some formulas custom soy as a source of protein and minerals. Minerals are essential for biological process due to their forming, bone mineralization, enzymatic reactions, secretion of natural hormones, cells, and lipid protection. The mineral content of formula milk has been added and fortified as in fulfilled desires of iron and zinc in formula milk. Macro and micromineral content in milk is an indicator of the quality of formula milk for infant growth and development. In this activity, the macro and micromineral content in infant formula are carried out by the neutron activation analysis method. An AA is a non-destructive multi-element sensitive and widely used technique for characterizing biological and food samples. An analytical purpose for infant formula milk has been observed previously. They were stated that the transfer of hazardous chemicals in the food chain due to the environmental pollution, such as in the manufacturing process. Therefore, it is necessary to monitor food safety in the Union of Europe. The metal in infant formula was calculated and estimated index according to the recommended level and could be tolerated for this element. This presents to the aim to determine the macro and micromineral composition using neutron activation analysis to compare the measured value with the corresponding nutritional label by manufacturers on commercial milk packs and to evaluate formula milk daily intake by considering daily nutritional requirements. And this is the methodology. There are 5 steps. The first one, sample collection. The sample collection are sample from infant formula milk and standard material for method validation. The second one for sample preparation, about 25 to 40 milligram infant formula milk weight put into 0.3 polyethylene vials and for SRM typical diet were used to validate the method. The third one, standard preparation. An AA standard was carried out by pipetting 100 microliters of ICP multi-element standard and irradiation and measurement gamma ray. The sample SRMs and mixed standard were irradiated with neutrons in reactor for 3 days. The irradiation exposure over a day and a week. Radionuclides was carried out by the high pure germanium multi-channel gamma ray spectrometers. And last one, calculation of estimated daily intake. Estimated daily intake of macro and micro elements for 6 months old infants were calculated based on the following formula. EDI. An infant's daily milk intake equal to 780 milliliter per day for 6 months old infant was assumed in the study. This is for result and discussion. Show that the method validation was carried out by testing SRM typical diet, which has a matrix similar with the sample and using the identical method to acquire a valid test method and guarantee the quality to the test result. The measurement result value of SRM and the certificate value is shown in the table. Certificate value and analysis result given relative bias in the range of 0.6 to 6.9%. And this is the figure 1, the ratio of certificate value and SRM typical diet analysis result. The result shows that there is an appropriateness between the certificate value and the analysis result through the method testing is valid and reliable for analysis on infant formula milk samples. This is the table 1, the concentration of macro and micromineral in infant milk formula by an AA method. There are 5 elements: potassium, sodium, iron, selenium, and zinc. This value is compared with the value stated on the manufacturer label product with a recovery 96 to 118%. The value of the measurement of result was compared with pure cow's milk without fortifying was carried out by Erwick et al. Using ICP-MS method. From this comparison show that the value of potassium sodium almost have similar concentration value. In contrast, the concentration of iron in formula milk shows 4 times higher than cow's milk. Meanwhile, zinc in cow's milk has greater content than formula milk. The selenium concentration in cow's milk is smaller than formula milk. The result of previous research conducted by Daniels et al. Analyze the content of macro and micronutrient elements in breast milk. Daniels said that breast milk is a sort of nutritional for infants up to 6 months of the recommended by WHO. Exclusive breastfeeding can be protected against gastrointestinal infections during the early growth of the infant. Macro and microelement can cure several problems such as clinical respiratory system, cancer, skin disorders, anemia, blood depression, reproductive disorders, heart failures, gastrointestinal disorders, fatigue, decreased immunity, and even death, which has consequences for public health. And this is the figure 2, daily intakes of macro and micro mineral from infant milk formula. Previous studies said the value of daily intake of formula milk conducted by Berlandaskan, iron, zinc, and selenium were obtained with EDI value 4.7, 4.9, and 0.6 milligram per day. This EDI value is different from the amount of the study. It was caused by the nutritional intake of formula milk requirements for each country is different and can be obtained by quantification substance contained in milk formula. After 6 months, the non-breast period of children usually consume baby formula milk, continuation formula milk, and various other supplementary foods, and another food intake such as vegetable food. Based on research conducted by Chekri, contributed 94% of the total macro and microelement concentration was obtained from food. Therefore, available food is an essential part of infant and toddler in evaluating the distribution of nutrients in the macro and micronutrient elements in the body. And this is the last part, conclusion. In the study, the value is still within the recommended intake set on the factorial value level. By consuming infant formula milk powder in adequate quantities, this commodity can for my presentation. Thank you so much for your attention.
Okay, thank you, Miss Indah Kusmartini, for the nice presentation. This is the end of the second group. So now we have a discussion session. In the chat room, we have one question from Miss Emma Damayanti. The question is for Miss Lia Ratnawati. What is the main component of antioxidant in ginger, and is it further analyzed the content of its antioxidant compounds? Is there any further analyzed the content of its antioxidant compound? Maybe the meanings. So please, Miss Lia Ratnawati, you can answer it.
Thank you, moderator. Thank you, Mrs. Emma, for the question. In this study, the main content of antioxidant from literature such as gingerol, paradol, and sogaol. But in this study, we just analyze the antioxidant activity.
Okay.
I think enough.
The next question, the second question. There are two questions, I think.
We don't analyze in this study. We don't analyze the antioxidant compound, but we just analyze the antioxidant activity.
Okay.
Yes.
Okay, Miss Emma.
Okay,
Is it enough?
it's enough.
Okay, thank you. So we go to the next question from Miss Emma again. I think you are the best questioner for this session. So the question is for Miss Indah Martini. The question is, what could possibly cause a bias value in the content between in the milk sample and in the label? So please, Miss.
Thank you very much for the question. In the previous research in my manuscript, in contrast, the concentration of iron in formula milk shown for time higher than pure milk. And also for zinc in cow's milk has a greater than formula milk.
Hello? Hello?
Can you hear my voice?
Very loud
Is it
voice.
clear?
Yeah.
I'm sorry. So,
Maybe it can cause from like sample number or storage of the milk, maybe.
Storage of the milk. Maybe one of the reason maybe for storage, but I think for the environment maybe it could be one of the reason of the difference between the concentration of macro and micro mineral in milk sample and infant formula milk, I think.
Okay.
Okay, enough, Miss Emma?
Okay, enough. Thank you.
Okay, thank you, Miss Kusmatini, for the answer. So is there any question? Okay, we got another question. In the chat room, Miss Lusti asked to Miss Indah Kusmartini again, does gamma irradiation of micro and macronutrients are common method for fortified mineral in commercial powder milk due to its lower content mineral during the drying process? And does the irradiation process also affect another parameters such as nutrient composition? Okay, you can answer it.
Thank you for the question. For gamma irradiation, mineral in commercial powder, the last previous research also analysis for another method like ICP-MS or spectrometer absorption of spectrometer. But in this research, the irradiation process, I think in the benefit of the irradiation process is we have analyzed selenium. I think for another method, sometimes selenium cannot be analyzed like that. And does the irradiation process also affect another parameter? nutrition composition, the irradiation method is focusly for the analysis of element. I think for the nutrition, like a vitamin, protein, there is no effect for the for the vitamin, protein, something like that. But this in the irradiation gamma rays is focused to analyze this element in powder milk.
Okay. Okay. Is it enough?
Yes.
Lucy?
Yeah,
Okay.
but I cannot hear the voice clearly.
Yeah. Maybe Miss Indah, you can put your speaker closer to your mouth. Okay. Next, your voice is quite very low. Okay, so we have another question from Miss Devi Desni Lasari to Miss Indah Kusmartini. Is it right?
Yes.
Oh yeah. About the method, could you explain more detail about neutron activation?
Okay, thank you. For the method, maybe the sample in formula milk powder is white. Also with standard and reference material, we prepare and we irradiate to the sample SRM and standard irradiation process for about 3 days in reactors. And we duration time required because the radiation exposure like potassium, potassium, and for medium half time, also for iron, selenium, and zinc, they have half time respectively. After irradiate in the reactors, the measurement element for radionuclide like potassium, sodium, we analyze by gamma ray spectrometer and Genie software. And after we calculate, we can calculate again the concentration, the gamma rays, and we can compare with the standard of the standard and SRM.
Hello, Miss Indah.
Is it clear?
Hello, Miss Indah.
Is it clear? Is it clear?
Okay, Miss Dewi Desnilasari, is it enough? Clear enough?
I think it's enough. Thank you.
Okay, thank you, Miss Indah, for the answer. And the next question is from Mr. Ahmad Sarifudin, also addressed to Miss Indah. What is the advantage of neutron activation method in mineral analysis compared to the established methods such as... Okay, you can answer it.
Yeah, thank you. For NAA, the benefit of nutrition analysis is non-destructive, multi-element, sensitive, and widely used technique for analysis biological. But in fact, the neutron activation analysis, you can analyze the element directly to what is the meaning of unsur inti. For I know for AAS, XRF, and etc. The analysis method the analysis direct to from the electron from the element, but for the activation analysis direct to how to say in the atom, yeah, direct to in the atom, like that. The benefit. So that's why the sensitivity of the method, we could say, is it more benefit than another method.
Okay, Mr. Ahmad Sarifudin, is the explanation clear enough? Or you want
Thanks.
to ask? Okay. Thank you, Miss Indah, for the answer. And we still have time for our discussion session. If you have another question, you can raise your hand because we don't have any question in chat room. So anybody, if you have question, you can raise your hand. No? No? Are you sure? No question again? Okay. Okay, actually we still have about 10 minutes. No, 5 minutes. 5 minutes, okay. Because no question anymore, so I think I will close this session. So, well, this is the end of our parallel session. And thank you for all presenters for the interesting presentation. And thank you for all participants who still being stay here until now. And please give a big virtual applause for the all presenters. And this is also at the end of our conference program for today, for the first day. And I would like to remind you that don't forget to join our second day tomorrow. There will be many great speakers we have for tomorrow plenary session. So please don't miss it. Okay. For this moment, I would like to close the program for today. Thank you again. Stay healthy and stay safe and see you tomorrow. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
See you tomorrow.
Thank you.
You can leave the room now.